Question number 51 says that in situ conservation, it basically refers to. So this is talking about one of the important strategies that has been mentioned over here regarding the biodiversity. And let me tell you that the conservation strategies are of two types. One is called as the in situ conservation, which we also called as the on-site conservation. All right. So biodiversity conservation. So biodiversity conservation is at two levels. Conservation is at two levels. So the first one is considered to be as the in situ conservation, which is the on-site conservation, which we already know about it. This is on-site conservation. That means the animals and plants, that means the organisms are protected in their natural habitat. So that's why it is called as on-site conservation. While the ex situ conservation is considered to be as the off-site conservation. That means that the organism is taken out from its natural habitat and is actually preserved, or you can say, and is actually taken to a different habitat, all right, where it is provided all with all those necessities and all those requirements that were available in its own natural habitat. So that's why it is called as on-site, and this is called as off-site conservation. Now, when we are talking about, since here it has been asked about in situ conservation, so when we consider in situ conservation, so this is going to protect and conserve the entire ecosystem on a whole basis. All right. So this basically protects and also conserves the whole ecosystem. So this is going to conserve the entire ecosystem or the whole ecosystem. All right. So it includes, like, national parks, wildlife sanctuaries, then biosphere reserves, all these categories. All right. So that's why the correct answer here will be option number A. And option B basically says that conserve only high risk species, conserve only endangered species, conserve only extinct species. So definitely these are not the option, because the entire conservation is done on a whole basis, because the entire natural habitat is being included. And if the entire habitat is included, then definitely the protection and conservation is taken care of on terms of the entire ecosystem. So option A is the correct answer. Now question number 52. Now, in which of the following animals the digestive tract has additional chambers like crop and gizzard? All right. So you have to now identify the categorizations has been given over here in the options given below. And you have to identify the digestive tract. That means the digestive area having this crop and gizzard. Now, let me tell you that basically crop is considered to be as a kind of a stomach part. All right. So this is going to crop is basically involves the storage compartment of the food, and gizzard is important for grinding of the food particles into smaller pieces. All right. And this is very prominent. That means this crop and gizzard, they are very prominent in case of birds. Gizzards are found in birds. So they're usually found in birds. So this is a kind of a crop, is basically a storage organ for food. Storage organ for food, while gizzard is a grinding organ, you can say. Grinding organ that helps in grinding food particles, converting or grinding food. All right, so this is the case, and since these are usually found in case of birds, so let us take a look that what are the different categories that has been given. This is the first important category is Corvus, Columba, and Chameleon. Now let me tell you that this Corvus, all right, Corvus is the scientific name of crow, all right. Then Columba, and this is a bird, all right. Then Columba is considered to be as pigeon. This is the scientific name of pigeon, all right. And again, this is also a bird category, all right. Then there is chameleon, I guess. So chameleon is basically a reptile category, all right. So chameleon is a reptile. This doesn't come on the bird category. All right. Now, so definitely this won't be an option because this is the two are birds, but the third one is the reptilian category. Then there is Bufo, Balanoptera, and Bangaris. So Bufo, Balanoptera, and Bangaris, if I tell you, Bufo is basically an amphibian. This is a category of frog, Bufo. So Bufo is an amphibian. Then amphibian. Then Balanoptera is an aquatic mammal. Balanoptera. Balanoptera. This is an aquatic mammal. All right. And then there is Bangaris. Bangaris is also a reptile. So Bangaris is also a reptile. So again, this is also not an option to be included. Then Catla, Columba, and Crocodilus. So Catla is basically a category of fish. All right. So Catla is a fish category. So definitely this is also not an option. Crocodilus is a reptile. We all know this is crocodile. So Crocodilus. This is a reptile. All right. So again, this is also not an option. And the last is Pavo, Pistacula. That is Cytacula and Corvus. So Pavo is basically peacock. Pavo is a category of peacock. Just a second. This is peacock. And this is a bird, of course. Then Cytacula is a category, or this is the scientific name of this one, parrot. And the last one, which is left over here, is Corvus. Corvus, we all know. Corvus, we have seen that Corvus is basically a category of crow. Here, that means this one, then this one, and this one. These are the birds category. This is also a bird. All right. So definitely this is the correct option. That means option number D is the correct answer. All right. Now, question number 53. So question number 53 says that if the length of a DNA molecule is 1.1 meters, then what will be the approximate number of base pairs that will be found? All right. Now let us just calculate this part. This is a calculative question that has been asked. So the formula, that means if we have to find out the length of the DNA, basically, length of the DNA, we'll have to find out the number of base pairs, basically. So the formula is like length, the entire length of DNA, it actually depends upon the number of base pairs. And along with that, what is the distance between two consecutive base pairs. So length of DNA molecule is equal to number of base pairs. So this is equal to number of base pairs multiplied it by distance between two consecutive base pairs. So distance between two consecutive base pairs. All right. So number of base pairs, we have to find it out. This we have to find it out. All right. Distance between two consecutive base pairs turns out to be as your, we know that the distance between two consecutive base pair is basically 0.3 it's 3.4 angstroms, 3.4 angstroms. All right, so if we just convert it into the respective length, if I say into respective meter, if I say, then that will turn out to be as, so we can say that the length of the DNA molecule is 1.1 meters. And you have to find out the number of base pairs. And the distance between two consecutive base pair turns out to be as 0.34 into 10 to the power. Since here the length is given in meters, so we will definitely write this distance between two consecutive base pairs in meters also. So like this. So if we just calculate the number of base pairs, therefore number of base pairs will be 1.1 divided by 0.34 into 10 to the power inverse 9. All right. Now this can be written as, once you calculate it, so this will be approximately coming out to be as 3.6 into 10 to the power 9 base pairs, which is almost equivalent to, if we just calculate it. So if we are going to see to it that what will be the condition, then if we just calculate it and if we just see to it, then this will turn out to be as 3.6 into 10 to the power 9 base pairs. So let us take a close look that it is approximate to what base pairs. So it's 3.3 into 10 to the power 9 base pairs. So this is actually coming in close. It is approximately coming in close with this 3.6. So definitely this can be the answer. But let us take a look in the other options as well. It's 6 into 610 base pairs, definitely not. 3.3 into 10 to the power 6 base pair, definitely not. Then 6.6 into 10 to the power 6 base pairs, no. So the only one that is coming in equivalent to this is 3.3 into 10 to the power 9 base pairs of DNA. All right, so that means option A is the correct answer. Now, question number 54. Now, question number 54 says that given below are the two statements, statement 1 and statement 2. Now, statement 1, with regard to these two statements, you have to choose the most appropriate answer from the options given below. Most appropriate answer you have to identify. All right. Now, if we just look into the statement 1, which says that fatty acids and glycerols cannot be absorbed in the blood. So definitely we can consider it to be a correct statement, because we know that these fatty acids and glycerol, they are actually insoluble in water. All right. And we know that our blood composition, 80% of our blood is primarily, 80 to 90% of our blood is primarily composed of water. The main constituent of our blood is water only. All right. So definitely these fatty acids and glycerol, they cannot be absorbed into the blood. All right. And so they are first incorporated into, they are actually converted into, smaller particles, and those smaller droplets are considered to be as micelles. All right. So we can say that this is a correct statement basically. Statement 1 is correct. Is correct. Now, why it's correct? Because fatty acids and glycerol. So fatty acids and glycerol being insoluble in water. So we know this fact that fatty acids and glycerol, they are insoluble in water. That's why cannot be absorbed in blood. That's why can't be absorbed in blood. All right. So that is the case. So specialized, now the statement second says that specialized lymphatic capillaries called lacteals carry chylomicrons into lymphatic vessels and ultimately into the blood. So this is also a correct statement. And the reason for this is because, as I told you, that once since these fatty acids and glycerol, they cannot be absorbed in the blood. So that's why they are converted into small droplets. So they are first converted into small droplets. So they are first converted into small droplets, which are called micelles. And once they are converted into micelles, this ultimately moves into the intestinal mucosa. So, which ultimately moves into intestinal mucosa, which is ultimately going to move into intestinal mucosa. And from there, once they are absorbed by the intestinal mucosal epithelium, from there they are reformed into smaller protein coated fat globules, which are called as chylomicrons. All right. So they are then reformed. They are then reformed into very small protein coated fat globules. So they're then reformed into very small protein coated fat globules, and that are called as chylomicrons. Those are called as chylomicrons. And these chylomicrons, they are ultimately transported into the lymphatic vessels. That means lacteals in the villi of the intestine. So these chylomicrons, these are ultimately transported into the lacteals. In the villi. And from there, once they are absorbed in the form of lacteals in the villi, and from there, these lacteals ultimately releases these absorbed substances into the bloodstream. All right. So these lacteals ultimately releases the absorbed substances in bloodstream. All right. So this is the case. So we can say that this second statement, that means specialized lymphatic capillaries called lacteals carry chylomicrons into the lymphatic vessels and ultimately into the blood, is a correct statement. That means both the statements are considered to be as correct. All right. So option A is the correct answer. Now, question number 55. So, question number 55 says that given below again, two statements are given, statement 1 and 2, and you have to choose the most appropriate answer from the options given below. All right. Now, statement 1 says that, just a second. Yeah. So statement 1 says that autoimmune disorder is a condition where body defense mechanism recognizes its own cells as the foreign bodies. All right. Okay. So autoimmune disorder is a disorder. It's a condition basically of this one, which is saying that autoimmune disorder is a condition where the body defense mechanism is considered to be the it's a defense mechanism which is actually considered to be a a disorder in which a condition where a body defense mechanism recognizes its own cells as the foreign bodies. So this is definitely considered to be as a correct statement, because autoimmune disorder, as the word is suggesting, autoimmune disorder, all right, that means definitely the immunity system will be affected. All right. And auto means self cells. That means when the body immune system is deficient enough, or the body defense mechanism is not able to recognize its own cells, and that's why it starts recognizing its own cells as any kind of a foreign bodies. So that's why it start attacking its own cells. So this might be due to genetic reasons or any other unknown reasons. That means because of which the body will start attacking its own cells. So definitely this is a correct statement. Basically statement 1 is correct. So statement 1 is correct. All right. So we can say that autoimmune disorder is a condition where body defense mechanisms recognizes its own cells as foreign bodies. Recognizes its own cells as foreign bodies. And so it will start attacking cells. And so the body start attacking cells. Cell cells. So this is a true statement. All right. Now the statement second says that rheumatoid arthritis is a condition where the body does not attack cells. Now the statement second is basically incorrect. This statement is considered to be incorrect because rheumatoid arthritis is an example of autoimmune disorder. And if it is an example of autoimmune disorder, then definitely it will fall into the category where the body will attack its cells. So statement 2 is incorrect, you can say. This is considered to be incorrect because rheumatoid arthritis, or you can rather say since rheumatoid arthritis is an example of autoimmune disorder, disorder. So body attacks cell cells. The body will attack cell cells. All right. So this is the case. So that means statement 1 is correct, but the statement 2 is incorrect. That means option D. Sorry, not option D. But if you say here, the statements are actually given in a proper incorrect way. So basically statement 1 is incorrect. So this won't be the case. Option D won't be the answer and option B won't be the answer. And option A says that statement 1 and statement 2 both are correct. Again, this won't be the case. So definitely only one option is left, and that is option C. Actually, the statement here is a little in a haphazard way, but it says that statement 1 is considered to be as correct and statement 2 is considered to be as incorrect. So statement 2 is incorrect, but the statement 1 is correct. So that means option C is the correct answer over here. All right. Now, question number 56. Now, question number 56 says that which of the following statements are true for spermatogenesis but do not hold true for oogenesis? Now, you have to identify that which is a statement that will be true for spermatogenesis but will not be true for the oogenesis. All right. So, let us take a look into the different options that has been given over here. So the first says that it results in the formation of haploid gametes. All right. So definitely in both spermatogenesis and oogenesis, if we will see. All right. So in both processes, we can say in both processes, haploid gametes are formed. That means sperm is formed in spermatogenesis and ovum is formed in oogenesis. So haploid gametes are formed. So definitely this statement is true for both cases. So basically we can say that A is true for both processes. All right. That means this cannot be the statement that we will have to include it, because we have to look out for that statement which is true for spermatogenesis but that is not true for the oogenesis, remember. So definitely this won't be the case. Then the second says that differentiation of gamete occurs after the completion of meiosis. Now, this is again very, very important statement, because the gamete differentiation occurs after the completion of meiosis. Now, in case of spermatogenesis, if you will see, so definitely it will after the completion of meiosis 1 and meiosis 2, this will result into sperm formation. So after, you can say that in spermatogenesis, meiosis 1 and meiosis 2. All right. That means after the completion of both the meiosis, after its completion, results into gamete formation. Results into gamete formation. All right, that means sperm. But in case of oogenesis, all right, in case of oogenesis, if we will be seeing, then the completion of meiosis doesn't differentiates the gamete, but it is because of the sperm entry into the ovum that induces the gamete formation. So in oogenesis, the sperm entry inside secondary oocyte. The sperm entry inside secondary oocyte induces completion of second meiotic division, that means meiosis 2, and then only the ovum is released. Then only ovum is released or formed, you can say, basically. All right. So definitely this is not true for oogenesis, we can say. So this is a true statement. Then there is the C statement says that meiosis occurs continuously in a mitotically dividing stem cell population. All right. So meiosis occurring continuously in a mitotically dividing stem cell population. So this is actually true. This is going to be true for the process of spermatogenesis, but this won't be true for the process of oogenesis, because once after this, once after these oocyte, that means the process of the division of the meiosis 1 and meiosis 2 in the oogenesis process occurs at a different stages of the entire life cycle. So definitely we can say that this is true for, that means statement C is true for C. Is true for spermatogenesis, but not for oogenesis. But not for oogenesis. All right, so this is also a true statement. Now, the next statement is that it is controlled by the luteinizing hormone and follicle stimulating hormone secreted by the anterior pituitary. Definitely, the entire process of spermatogenesis is controlled by only these two hormones. That means luteinizing hormone and follicle stimulating hormone. Luteinizing hormone is basically, these, and let me tell you that these hormones, all right, so and both the process, actually, not only the LH and FSH is being controlled, is controlling the process of spermatogenesis, but both these hormones, they play a very important role in oogenesis also. That means this statement is actually true for both the processes. All right, because this luteinizing hormone uh, is basically being released by the interstitial cells or the Leydig cells. All right, and this one, that means luteinizing hormone or the LH surge in case of the oogenesis is responsible for the process of ovulation. All right. So basically, this luteinizing hormone is responsible for acting on the Leydig cells and thereby releasing the testosterone hormone in spermatogenesis. And follicle stimulating hormone is responsible for maturation of the follicles. All right. So indirectly, we can say that both of these hormones, they are going to be true. They are very, very essential hormones that is required in both the processes. So definitely, this cannot be considered to be as the statement, the correct statement. So we can say that LH and FSH plays very important role, very important roles in both processes. So since we only have to go for, look out for that process which is true for only spermatogenesis but not for oogenesis. So this thing you have to keep in mind. All right. And the last is that it is initiated at puberty. All right. Spermatogenesis, now let me tell you, definitely this process is initiated at puberty. So spermatogenesis begins at puberty only. Spermatogenesis begins at puberty. So since this process begins at puberty, that means at an age of 12 to 14 years of age. All right. due to sexual maturity and release of various kinds of hormones. While the oogenesis process, this begins during the embryonic development, all right, of the female. So oogenesis begins at, or begins during, you can say, during embryonic development of female. All right. So definitely this is a statement which is not true, which is true for spermatogenesis, but not true for oogenesis. So this is also correct statement. So if we have to choose the most appropriate answer from the options given below, then option B, C, and E is the correct answer. That means option D over here. All right, now question number 57. Question number 57 again says that given below are the two statements. One is labeled as assertion and the other labeled as reason. So you have to find out that whether the statements are correct or whether the reason is correct for the assertion or not. So osteoporosis is characterized by decreased bone mass and increased chance of fractures. So definitely this is a true statement, because osteoporosis is caused because of decreased level of estrogen hormone, and definitely this results into decreased bone mass and increased chances of fracture. So A is correct. That means the assertion is correct, because osteoporosis is characterized by decreased bone mass and apart from that, increased chance of fractures. Chance of fractures also. And this is because both these factors, this is because of decreased level of estrogen. This is due to decreased level of estrogen hormone. That's why. All right. Now let us take a look into the reason. So the reason is that the common cause of osteoporosis is the increased level of estrogen. No, the reason is incorrect. All right. That means R is actually not incorrect, because it is because of the decreased level of estrogen and not because of the, decreased, not because of the increased level of estrogen, but it is because of the decreased level of estrogen. All right, all right. So definitely the reason is incorrect. So R is incorrect. So let us take a look. Both A and R are correct, and R is the correct explanation. No. Both A and R are correct. Again, no. Then A is correct, but R is not correct. Option C is the correct answer. A is not correct, but R is correct. Again, no. All right, that means option C is the correct answer. Now, question number 58. So question number 58 says that given below again are the two statements, and you have to choose the most appropriate answer from the options given below. All right. Now, this says that restriction endonucleases recognizes specific sequence to cut the DNA known as palindromic nucleotide sequence. All right. So definitely this is a correct statement, because restriction endonucleases are those nucleases that recognizes a specific site, and that specific site of sequences is considered to be as a palindrome sequence only. All right. So you can say that statement 1. So statement 1 is correct. Why it's correct? Because this is one of the natures of restriction endonuclease, that restriction endonucleases are the restriction enzymes are the restriction enzymes that recognizes specific site remember specific site to cut DNA. All right, and those specific sites are considered to be palindromic. These are these specific sites are called as the restriction site, you can say, or recognition site, you can say. So these sites are known as restriction sites or palindromic sequences. That means the sequence, if you will read it out from both the left and right side, it will turn out to be similar. So palindromic sequences. All right, palindromic sequences. Clear? Now, option, that means statement 2 basically says that restriction endonucleases, it cuts the DNA strand a little away from the center of the palindromic site. Now, again, so this is also correct statement that it is not going to cut the strand at the center, from the center of the palindromic sequence or at the center of the palindromic sequence, but little away from the center, because this will be between the same two bases and that will be on the opposite strand. So we can say that statement 2 is also correct. So statement 2 is correct. Statement 2 is correct because restriction enzymes cuts the strand of DNA a little away from the central center of palindromic site. Palindromic site, but between the same two bases on both the strands. Same two bases on the opposite strands. All right, so this will be the reason. So that means both the statements are considered to be correct. So statement 1 and statement 2 both are correct. Again, this is a correct statement. The first has turned out to be again, so definitely these statements are wrong. All right, so option A is the correct answer. Question number 59. Now, question number 59 is asking about lipase loop. That lipase loop is a type of, all right. Lipase loop is a type of contraceptive used as. Now, let me tell you that a lipase loop is basically a category of non-medicated IUD. All right, so basically IUDs are of three kinds. All right, IUDs which are considered to be as intrauterine devices. So intrauterine devices, and these are primarily used only for females. So these are primarily three different categories. It can be medicated IUDs. It can be it can be non-medicated IUDs, then hormone-releasing IUDs, and copper-releasing IUDs. So three categories: non-medicated IUDs, then hormone releasing IUDs, and then there is copper releasing IUDs. Copper releasing IUDs. So in case of non-medicated IUDs, it includes lipase loop, the only one category example that has been given in your books also. Hormone releasing IUDs includes Progesta Cert and LNG20. It's Progesta Cert and LNG20. And copper releasing IUDs includes Copper T, Copper 7, and multi-load 375. Multi-load 375. All right, so definitely lipase loop is a category of non-medicated IUDs. While the other barriers, if you will see, then cervical barrier and vault barrier. Cervical and vault barrier, these are a category of physical barrier. These are the basically diaphragm walls and cervical caps. These are these are another categories of the physical barrier only, which are used in case of females. All right, so cervical barriers and vault barrier. So cervical barrier and wall barriers, these are physical methods of contraception. These are barrier methods. And also diaphragms you can consider. Diaphragms. Physical barriers or methods of contraception. So definitely the correct answer is option C. All right, now question number 60. So question number 60 says that under normal physiological conditions in human being, every 100 ml of oxygenated blood can deliver dash ml of oxygen to the tissues. All right. So if you just consider the normal conditions, and it is asked, since it is asking about the normal physiological conditions, because if we consider regarding the deoxygenated blood, let me tell you that every 100 ml, all right, so every 100 ml, this is a fact, that every 100 ml of deoxygenated blood, since here it is asking about oxygenated blood, but every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. To the alveoli. All right. So definitely, if we just consider the normal conditions regarding the case, so in every normal conditions, if we have to see it, so the more closure proximity, if you are going to see to it, all right, then every 100 ml of oxygenated blood is basically responsible for delivering around 5 ml of oxygen to the tissues under normal physiological conditions. All right, so this is a fact and also a statement that has been given in your books as well. So that's why the correct answer will be 5 ml over here. All right, so we can say that every every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to tissues under normal physiological conditions. Normal physiological conditions. All right. So that's why option B is the correct answer. So question number 61. So question number 61 says that given below are the two statements, and one is basically labeled as assertion and the other labeled as reason. All right. And you have to go through the assertion and reason. And in the light of the above statements, you're supposed to choose the appropriate answer from the options given below. All right. So that is the case. Now let us take a look that what are the different assertion and reason that has been given. Basically, the assertion says that all vertebrates, they are chordates, but all the chordates, they are not vertebrates. All right. So the reason that has been given for this is notochord is replaced by vertebral column in the adult vertebrates. Now, let me tell you that both the statements that has been given, which is assertion and reason, actually is correct. All right, because chordates, if we just talk about the chordates category, then chordates are primarily divided into three subphylums. So chordates are divided into three subphyla, or three subphylum, you can say. All right, so they are primarily divided into three subphylums. The first one is called urochordate or urochordata, then cephalochordata, and then vertebrata. All right. So urochordates, cephalochordates, and then vertebrate. Now, in this category, that means in the vertebrate category of the chordates, they have a presence of notochord, but this notochord is found only in their embryonic stage. However, this notochord is replaced by a complete and a full-fledged vertebral column in the adult stage. So, in this category, you can say, in this category, notochord is found in, so notochord is usually found in early or embryonic stage. All right. While it is replaced by, well, this is actually replaced by a cartilaginous or bony. Either it is cartilaginous or it is bony vertebral column. So vertebral column in adult stages, or you can say in the adult. All right. So therefore, we can say, and these two organisms, that means these two categories of urochordata and cephalochordata, they only have the presence of notochord. So they have only notochord. They have only notochord from early till adult stage. So till adult stage. So they have only this means notochord, while the vertebrata, they have notochord in the early stages, but it is replaced by vertebral column in the adult stages. So by this thing we can say, and by this statement we can say basically that all vertebrates can be considered to be as the chordates. All right, but all chordates, that means all the three categories, then they cannot be considered to be as vertebrate, because these two, that means urochordata and cephalochordata, they are not having vertebral column. So definitely they cannot be included, or they cannot be called as vertebrates. So therefore we can say that all chordates are vertebrates. Therefore, we can say that all chordates are all vertebrates are chordates, basically. Not all chordates are vertebrate. Just a second. All vertebrates. We can say that all vertebrates are chordates. All vertebrates are chordates, but all chordates are not vertebrates. All chordates are not vertebrates. All right, so this is the case. So with this, we have come to know that basically both the reason, that means both the assertion and the reason, it is considered to be as correct. And in fact, the reason is said to be as the correct explanation of the assertion. So that's why option A is the correct answer. All right, now question number 62. Now, this question says that which of the following is not a connective tissue? All right, so we have to find out the category which is actually not a connective tissue. So, blood, the options that has been given, that is blood, then there is adipose tissue. So, blood, adipose tissue, then cartilage, and neuroglia. All right. So these are the options that has been given. All right. So let me tell you that these three categories, all these three categories, that means blood, adipose tissue, and cartilage, they are considered to be as the connective tissue, or they are a category of connective tissue. All right. So blood, adipose tissue, and cartilage, they are actually a category of connective tissue. And that too, if I tell you, then this blood and cartilage, blood and along with cartilage, they are a category of specialized connective tissue. So these are specialized connective tissue, while connective tissue. While on the other hand, the adipose tissue is a category of loose connective tissue. So adipose tissue is an example of loose connective tissue. So this means all these three options cannot be the correct answer, definitely. So the only option left is the neuroglia. All right. So neuroglia is considered to be as the correct answer, because neuroglia is basically this is a part of the nervous system. All right, or neural tissue, you can say basically. And these are the supportive cells that makes up the nervous tissue, and it almost makes up, half of the volume of the neural tissue. So neuroglia is a part of nervous tissue. So this is a part of nervous tissue that supports it. So that is going to support it, and they make up almost. This is actually considered to be as making up the volume of, more than half of the neural tissue. So that's why they make up. They make up more than half of the volume of neural tissue. All right. So that's why the correct answer is option D over here, and that is neuroglia. All right. Now, question number 63. Now, question number 63 says that nitrogenous waste is excreted in the form of pellet or paste by. All right. So the options that has been given is basically ornithorhynchus, then salamander, then hippocampus, and then pavo. Now let us take a look into the different options that has been given. So the first option which is given over here is ornithorhynchus, and ornithorhynchus is basically the duck-billed platypus. All right, and this is a category of mammal. And mammal, they usually their excretion is usually in the form of urea. All right, and that's why they are considered to be as ureotelic animals. So definitely this won't be an option. So ornithorhynchus, which is also considered to be as duck-billed platypus. All right, so this is a mammal. So this is a mammal, and its excretory product is urea. Its excretory product is urea. So it is a ureotelic animal, basically. So definitely this cannot be an option. All right. Then option B is salamandra. Salamandra is basically amphibian category. All right. And salamander and hippocampus. Basically, hippocampus is a category of bony fishes, and both are of ammonotelic nature. That means they usually secretes excretion through ammonia, in the form of ammonia, basically. So salamander or salamandra and hippocampus. Which is a bony fish. And this is an amphibian. All right, aquatic amphibian. These both are considered to be as, both are ammonotelic. That is excreting ammonia. All right. The only option left, this is the B option and this is the C option actually here. So the only option left is option D over here, which is pavo. So pavo is a category of bird. All right. And this pavo, basically releases nitrogenous waste in the form of paste, and that paste is considered to be as uric acid. All right, so that since this is released in the form of paste, so that minimal water loss occurs through excretion. So that is the only reason. So that means power will be the correct answer. So it's a bird that excretes uric acid. So this basically excretes uric acid in the form of pellet or paste. All right, with a minimum loss of water. Clear? So the correct answer is option number D. Now, question number 64. So question number 64 says that regarding meiosis, you have to identify the incorrect statement. That which of the following statement is incorrect regarding meiosis it's talking about. So there are two stages in meiosis. That means meiosis 1 and meiosis 2. Definitely this is a correct statement. Option A is correct. So this is a correct statement. So it is correct because meiosis is divided into two stages. Meiosis divided into two stages. That is meiosis 1, which is considered to be, of course, the reductional division stage, and meiosis 2 is just the equivalent to mitosis. So that's why it's considered to be as equational division. So meiosis 1 and meiosis 2. So this is correct. Then in S phase of meiosis, the DNA replication, in S phase of meiosis, the DNA replication occurs. All right. Or meiosis 2 it is talking about. Actually, the statement is little haphazard. But let me tell you that the statement is saying that in meiosis 2, statement B says that in meiosis 2, or you can say in S phase of meiosis 2 rather. In S phase of meiosis 2, DNA replication occurs. This is the statement. DNA replication occurs. Now let me tell you that this S phase is basically the synthesis phase, which is DNA synthesis phase. All right. And DNA synthesis phase is a phase of interphase. That means it's a phase that comes, that is not included in the process of cell division. All right. It is a cell cycle event that comes under interphase, but not in the division phase. So DNA synthesis phase. S phase is a DNA synthesis phase. So this is actually the incorrect statement because this process of DNA replication, this process actually occurs in interphase stage, S phase of interphase, but not of meiosis 2. So this occurs in S phase of interphase and not meiosis 2. So definitely this is the wrong statement. That means the incorrect statement. So since we have to find it out, so this is definitely the right answer. But let us take a look into the other options as well. So this next statement says that pairing of homologous chromosomes and recombination occurs in meiosis 1. Again, this is a true statement because, it is correct. This statement is also correct because this pairing of homologous chromosomes and the recombination Pairing of homologous chromosomes occurs in the zygotine stage, and the process of recombination occurs in the pectine stage of meiosis 1. It is correct because pairing of homologous chromosomes occurs in zygotine phase of meiosis 1. All right, M1, while recombination occurs in pectine stage of meiosis 1. So this is a correct statement. All right. Now the last statement says that 4 haploid cells are formed at the end of meiosis 2. This is also a correct statement. All right. Because at the end of meiosis 2, all right. So it is also correct. It is also correct because this is because during the start of a cell like this, all right. So if a cell is having diploid chromosome, so this is going to divide and give rise to 2 cells like this. So this is in meiosis 1 I'm talking about. All right. So this is going to give rise to 2 cells and which are haploid like this. And when it will undergo the process of meiosis 2, all right. So this will undergo the process of meiosis 2 over here. So since this is an equational division, so definitely it is going to give rise to 4 cells, 4 haploid cells like this. N, 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 and N. So this means 4 haploid cells formed at the end of meiosis 2. All right, so this is also correct statement. So that means the incorrect statement is option B. Now, question number 65. Question number 65 says that in the E. coli strain of in an E. coli strain, the I gene gets mutated and its product cannot bind to the inducer molecule. All right. So if the growth medium is provided with lactose, then what will be the outcome? All right. So now we know this fact very well. So we'll have to figure it out with the different options because this is the question that has been asked from the lactoferrin gene. All right. So we know that there is an inducer which is found. There are 3 structural genes which is found in the lactoferrin. Then there is there is an operator binding region also. So there are these things. So since here basically as I gene is basically responsible for binding or binding to the operator site, because it basically releases a molecule that is responsible for binding to the that is responsible for binding to the operator region. All right. So here it is already given. The given statement is all right. All right. So basically, since in the given statement, the gene I has been mutated and its product cannot bind to the inducer molecule. So we will first have to draw a sample lac operon structure so that you people can have an idea. So if you just draw it, then a lac operon basically has a unit like this. So here is the promoter, and here is the inducer. Again is the promoter, then here is the operator, and this is Z, Y, and A. Z, Y, and A. So these we all know is considered to be Z, Y, A. These are basically the structural genes over here of the lac operon. This is the lac operon that and these are structural genes. Now we know that the function of the this one, that means the inducer molecule, is to produce a repressor protein. All right. So basically, this inducer gene, through the process of translation, so through the process of translation, this is going to produce a repressor mRNA. All right, repressor mRNA. And this repressor, this is not through the process of translation, but it will actually be transcription process, not translation. This is transcription process. All right, over here. Then with the help of translation process over here, this will be producing the repressor protein. Repressor protein. And the function of this repressor protein is to bind to this operator region. This is the repressor protein actually, which is going to bind to this operator region. So binds to operator. And once it will be binding to operator, there will be no transcription of the structural genes. They won't be transcribed or translated. All right. So this condition I am explaining if there is absence of inducer. In absence of inducer, I'm talking about. In case of absence of inducer, and in case of the absence of, yes. Yeah, just a second. So this I'm explaining you in the absence of the inducer, which is lactose. All right. And if there is absence of inducer, then this repressor protein will be binding to the operator region. And so all these structural genes won't be transcribed and translated. So these genes, Z, Y, and A genes won't be transcribed. Sorry, it's just a second. Won't be transcribed and translate. All right. So this is an absence of inducer I'm talking about. But when there is a presence of inducer, so there won't be the binding. That means if there is a presence of inducer, then this repressor protein won't be binding to the operator region, because that inducer, that means the lactose, will be further binding with the this one, that means the repressor protein. So let us just draw that part as well. For example, in presence of inducer, if we are considering in presence of lactose. So if there is presence of lactose, then again we are going to draw this part like this. Here will be promoter, then I gene, then promoter, operator, and Z, Y, A. These are the structural genes that are there. All right, so that is the case. So through the process of transcription, this I will be producing repressor mRNA, and this will be producing repressor proteins. Now we know that there is a presence of lactose over here. All right. So there is a presence of lactose molecule like this. And if there is a presence of lactose molecule, then this lactose molecule, definitely that means inducer, which is also called as inducer. All right. So this lactose molecule will be binding to the repressor protein like this. So this will be binding like this. All right. So this is inducer binding to the repressor protein. And once it binds it, it will convert the active repressor protein into inactive repressor protein. Inactive repressor. And once this repressor protein will be inactive, so this inactive repressor protein won't be able to bind to this operator, definitely. So this won't be able to bind to the operator region. And when this won't be able to bind to the operator region, so all these structural genes over here, they will be transcribed also and translated also. So all 3 genes are free to be transcribed and translated. Are free to be transcribed and translated. All right, so this is the case. Now let us move on to the question. The question was saying that I gene has been mutated, remember. So since there is a mutation in the I gene, so since I is already mutated over here, all right, if we are considering it, so since I gene is already mutated over here, so definitely it won't be able to bind to the inducer molecule, and its product cannot bind to the inducer molecule. And if growth medium will be provided with inducer, that means the lactose, then definitely all the genes will be translated. You can see if it won't be able to bind to the operator molecule, then definitely all the genes are free and they will be transcribed and translated. Clear? So we can say that here we can also say that they will be producing, first they will be producing the lac mRNA. Through the process of transcription, they will be producing lac mRNA. All right, and this lac mRNA will be further producing all the genes is specific. No? That's why. That means Z stands for beta galactosidase. It encodes for beta galactosidase enzyme. Then Y for permease and A for transacetylase. So option C is the correct answer. Clear? Now, question number 66. Now, question number 66 says that which of the following is considered to be as a correct match for the disease and its symptoms? It's talking about, correct match for the disease and its symptoms. Let us take a look into the options that has been given. So the first option is arthritis, and arthritis have the presence of inflamed joints, that is inflammation in the joints in the arthritis. So this is actually true. The first statement has turned out to be true. The first option has turned out to be true. So in arthritis, there is inflammation in joints. So there is inflammation in joints. All right. So this is actually a correct match, but let us take a look into the other options as well. The next option says that tetany is basically high calcium level causing rapid spasm. Now, let me tell you, it is because of this tetany results into rapid spasm, but it is because of the low calcium level in the body fluid. It is not because of the high calcium level, but it is because of low calcium level. So, tetany is caused because of low calcium levels in blood fluid, sorry, in body fluid, not blood fluid, body fluid, in body fluid, causing rapid spasms. All right, so definitely this is not a correct one. So option C says myasthenia gravis. Now this is a genetic disorder resulting in weakening and paralysis of the skeletal muscle. Now this is also considered to be actually incorrect, because myasthenia gravis is a category of autoimmune disorder. All right, and since it's an autoimmune disorder, so this is going to weaken, or this is going to affect the neuromuscular junctioning, leading to, you know, symptoms like fatigueness or weakening and paralysis. All right, so again this is also incorrect. So we can say that myasthenia gravis. So myasthenia gravis is an autoimmune disorder. Where the body will start attacking cells due to defective immune system. So it's an autoimmune disorder affecting neuromuscular junction autoimmune disorder affecting neuromuscular junction, leading to fatigue, weakening, and paralysis of skeletal muscles. All right, now, so the last is muscular dystrophy. Now, muscular dystrophy is an autoimmune disorder causing progressive degeneration of the skeletal muscle. Again, this is also incorrect, because muscular dystrophy is a genetic disorder. This is not an autoimmune disorder actually. So that's why this is incorrect. So this is, just a second, muscular dystrophy. So this is actually a genetic disorder. So a genetic disorder caused due to degeneration of skeletal muscles. All right. So again, this is also not the correct answer. That means the correct answer is this one, which is option number A. Now, the next is question number 67. Now, question number 67 says that identify the microorganism which is responsible for the production of an immunosuppressive agent. All right. Now, basically, immunosuppressive agent, and that is immunosuppressive molecule cyclosporin A. The name has been given, which is cyclosporin A. So let me tell you that cyclosporin A is basically a bioactive molecule. And this is a bioactive molecule which is produced by Trichoderma polysporum, which is a fungus category, a fungal species actually. So this is a direct question that has been asked, so we can like do it. This is the correct answer will turn out to be as option number A, Trichoderma polysporum. So you can say that cyclosporin A. This is a bioactive molecule produced by Trichoderma polysporum. So this is a fungal species actually. All right, is used as immunosuppressive agent in organ transplant patients. In organ transplant patients. All right. While on the other hand, if you see the different options, so Clostridium butylicum is a bacterial species which produces butyric acid. Clostridium butylicum. So Clostridium butylicum. So this produces butyric acid, which is actually an organic acid, but not a bioactive molecule. And then there is Aspergillus niger, I guess. Yes. So there is Aspergillus niger. So Aspergillus niger, this produces citric acid. So this produces citric acid. And the last is Saccharomyces cerevisiae. It is basically Streptococcus cerevisiae. So I guess it should be there, Saccharomyces cerevisiae. But this is a species, again, this is a species which is considered to be as brewers and bakers yeast. It should be here Saccharomyces cerevisiae, not Streptococcus cerevisiae actually. All right. So definitely these are not the correct option. That means option A is the correct one. All right. Now, question number 68. Question number 68 says that given below are the two statements, statement 1 and statement 2. Now, in the light of the above statements, you have to choose the most appropriate answer from the options given below. All right. Now, statement 1 says that mycoplasma can pass through less than 1 micron filter size, and mycoplasmas are the bacteria with cell wall. All right. Now, let me tell you that mycoplasmas, first of all, let me tell you that mycoplasmas, these are not the bacterial species. All right. Initially, they were put into bacterial species, but because of lack of cell wall, they have been excluded from the bacterial category. So definitely mycoplasmas, these are the smallest living cells known. Smallest living cells known. Living cells known. Lacks cell wall. Survives without oxygen. Survives without oxygen. So definitely the statement 2 is incorrect. But what about the statement 1? So as as the statement here has already been written that they are the smallest living cells to be known on this earth. So definitely they are the smallest cell, which are only 0.3 micrometer in length. So 0.3 micrometer in length. All right. So definitely it can pass through less than 1 micrometer filter. So this statement is also correct. This statement is correct, but this statement is false. That means statement 1 is correct and statement 2 is incorrect. That means option C over here. All right. Now, question number 69. Now, question number 69 says that which of the following functions is not performed by the secretions from the salivary glands? All right. So salivary glands, doesn't perform the following functions actually. So the very first important function is that the option that has been given is that it controls bacterial population in the mouth. So definitely this is true because saliva is an antibacterial agent. It has antibacterial enzymes present in them, which is lysozyme. So option A. So this is actually correct. We have to find out not performed. All right. So salivary glands produces saliva or secrete saliva that has antibacterial enzyme. And that antibacterial enzyme is lysozyme. So definitely this is considered to be as a correct statement in mouth, we can say. So this is a correct statement actually. Now option B says that this is actually incorrect with respect to, you can say, this is incorrect with respect to the not performed secretion. All right. So that's why. Then option B says that the digestion of the complex carbohydrate, definitely. The digestion, the digestion of the complex carbohydrates, it starts from the mouth itself. All right. Yes, it starts from the mouth itself, and this is because of the secretion. That means, and that too, let me tell you that the digestion of starch, basically, it starts from the mouth itself, which is the complex carbohydrate. So again, this is also true. The digestion of complex carbohydrates like starch is done by secretions from salivary glands. All right. So this is also correct actually. This statement is also correct. This statement is also correct. Then lubrication of the oral cavity, definitely. The saliva helps in lubricating it, lubricating the oral cavity or the mouth part, so that the process of chewing, the process of engulfing the food particle becomes easy. And this is because one of the main important component of saliva, which is released by the salivary gland, is mucus. So saliva, you can say saliva secreted by salivary glands. Salivary glands. Just a second. Saliva secreted by salivary glands have mucus. So since they have mucus, which helps in lubricating oral cavity. All right, so which helps in lubricating oral cavity. And the last is digestion of disaccharides. So definitely digestion of disaccharides is not carried out. This is not a correct statement. That means this activity is actually not performed by the salivary glands, because the digestion of disaccharides is actually carried out in the small intestine, not in the mouth. So digestion of disaccharides are performed in small intestine and not in mouth. All right. And that too from the secretions of the saliva. So salivary gland will only secrete saliva, and that will be in the mouth only. So that's all. So that means the correct answer with regard to this statement, if you have to find it out, not performed. So that means this is the correct answer over here. These are basically the wrong statement. We can say like this actually, but this is the correct answer, not performed, since here it is asking not performed. That's why. All right. Now, question number 70. Now, question number 70 says that again, given below are the two statements, and you have to identify the appropriate answer from the options given below. So the statement one says that the coagulum is formed of network of threads called thrombins, and spleen is the graveyard of erythrocytes. All right. Now, let me tell you that the statement one, which is saying that the coagulum basically means the clot, blood clot. So the blood clotting is basically formed because of a network of threads, and those are called as fibrins, not thrombins. That means fibrinogens. All right. So we can say that statement one is incorrect. Now, why it is incorrect? Because coagulum or clot is formed by network of threads called fibrins, but not thrombins. So definitely this statement is incorrect. But the second statement, which says that the spleen is the graveyard of erythrocytes is basically RBCs, and this statement is true. All right. So statement 2 is correct. That spleen is graveyard of RBCs. Graveyard of RBCs, as RBCs are destroyed over here. Destroyed here. All right. That means statement 1 is incorrect, but the statement 2 is correct. So let us take a look. So option D is the correct answer. All right. So let's talk about question number 71. All right. So this question says that a dehydration reaction links the two glucose molecules to produce the maltose. All right. So if the formula of glucose is C6H12O6, then what is the formula of maltose? All right. So here it is basically asking about the formula of maltose, that we have to find it out. All right. And a statement has also been given, in which it has been written that it is a process of dehydration reaction that will be producing maltose. All right. Now let me tell you that dehydration is basically one such process in which there is elimination of water. All right. So dehydration process Dehydration process involves elimination of water molecule. All right. So this is basically a process in which the water molecule is ultimately being eliminated from a particular uh, product, or you can say from a particular molecule, actually, elimination. So since we know that maltose is basically a disaccharide molecule. All right. So maltose is a disaccharide which is actually formed by combining two monosaccharides. So it's a disaccharide molecule formed by two glucose molecules. That means when two glucose molecules they combines to one another, then they will be forming a maltose molecule, and they ultimately form a glycosidic bond in between this molecule. All right. So how the process is being carried out? So we know that glucose. So we can say that glucose into glucose will give rise to maltose. All right. So here, since it is talking about the dehydration processes being involved, so glucose is represented as C6H12O6, and there will be again C6H12O6. So both will be ultimately combined, involving the process of dehydration. That means elimination of water molecule. So if I just eliminate the water molecule over here, so this is going to turn out to be, the formula will turn out to be C12. You can see there are 12 carbon molecules. Then H24 will be there. All right. But here, since it is involving the elimination of two hydrogen molecules, so here it will be finally H22, and then O oxygen molecules will be 12 over here. But since there is elimination of one oxygen molecule, so it will be O11 over here. All right. So this is primarily the maltose, the molecular formula of maltose, which is C12H22O11, formed as a result of the dehydration process in the glucose molecules, two glucose molecules. All right, so that's why the correct answer, if we just find it out, then the correct answer is option C over here. All right, now next is question number 72. Now, question number 72 says that which of the following statements with respect to the endoplasmic reticulum is incorrect? So, basically, certain statements are given below, and you have to find out the incorrect statement, and that too with respect to ER, endoplasmic reticulum. So, let us take a look in all the statements. So, option A says that rough endoplasmic reticulum has ribosomes attached to ER. Definitely, this is a true statement. All right, so this is a correct statement because rough endoplasmic reticulum has ribosomes attached to it. Ribosomes attached to it, and it performs protein synthesis. Performs protein synthesis. That means this is a correct statement. Now, option B says that SER, that means smooth endoplasmic reticulum, is devoid of ribosome. This is again also a correct statement. Okay. So basically, SER lacks ribosome. So this is also true. Then option C says that prokaryotes have, in case of the prokaryotes, if you're going to see, only RER is present. All right. So let me tell you that this is actually the incorrect statement, because in case of prokaryotes, the membrane bound cell organelles are actually absent. and ER is considered to be as a membrane bound cell organ. All right. So since this is a membrane bound cell organelle, so definitely in case of prokaryotes, neither RER is present nor SER is present. So this is an incorrect statement because in prokaryotes, all membrane bound cell organelles are absent. It is not only ER, but chloroplast, mitochondria, vacuole, lysosome, all these are absent. So all membrane bound cell organelles are absent. So all membrane bound cell organelles are absent, and so is the ER, which is also absent. All right, so neither RER is present nor SER is present. All right, so this is, we have found out our incorrect statement, but let us take a look into the last statement also, that SER are the sites for lipid synthesis. So yes, this is a correct statement that SER is primarily involved in the synthesis of lipids. All right, so basically we can say that SER functions in lipid synthesis. So this is a correct statement. So that means since we have to find out the incorrect statement over here, so that means option C is the correct one. All right. Now, question number 73. Now, question number 73 says that which of the following is present between the adjacent bones of the vertebral column? So this is basically asking about that which of the following tissue or muscle or category is found between the adjacent bones of the vertebral column. Now let us take a look into the different options. So the first option says intercalated disc. So basically let me tell you that intercalated disc is one such category of cardiac muscle. It's one such category that is actually uh, found, or this is a characteristic feature of the cardiac muscles, and that is present in the heart. So it definitely cannot be present between the adjacent bones of the vertebral column. So that means this is considered to be as incorrect. All right, because intercalated discs are present in the heart. And is a characteristic feature of cardiac muscles. So this is basically a characteristic feature of cardiac muscles. This is incorrect. Then option B says cartilage. All right. Now cartilage is considered to be as a correct answer because this cartilage, they form a kind of an intervertebral disc that is found between the adjacent bones of the vertebral column, because and that category which forms, that is present between the adjacent bones of the vertebral column, is the cartilaginous joint. So basically, this is correct. This is a correct statement, correct option rather. So we can say that cartilage forms intervertebral disc between the adjacent bones of vertebral column. Vertebral column and is a type of cartilaginous joints. All right, so that means this is a correct option. Now let us take a look into this option C. Basically, see, option C, it has been written areolar tissue, and let me tell you that this tissue is a category of loose connective tissue which is present beneath the skin. So it has nothing to do with the presence of adjacent bones in the vertebral column. So definitely this is also incorrect. That means areolar tissue is a type of loose connective tissue. So this is basically a type of loose connective tissue that is found or that is present beneath the skin. So present beneath skin. All right. And the last is smooth muscles. So smooth muscles, basically again, these are present in the visceral organs. All right. That means the blood, the organs which are richly supplied with blood. So they are found over there. So again, they have nothing to do with the adjacent bones or their presence between the adjacent bones or vertebral column. So this is also incorrect. So smooth muscles are present in visceral organs. So definitely the correct answer is option B over here. All right. Now let's talk about question number 74. Now question number 74 says that tegmina in cockroach arises from. All right. So tegmina, it is asking about tegmina. Now let me tell you that tegmina is also considered to be as forewing. All right. So forewing, and that too, tegmina is considered to be as the first pair of wings, because if you just consider the different options that has been given over here, like prothorax, meso, and the thorax of the cockroach is divided into three categories: prothorax, mesothorax, and metathorax. Let me tell you that body of a cockroach is divisible into three parts. All right. So the first one is head, then thorax, and the third one is abdomen. And this thorax is further divided into prothorax, mesothorax, and metathorax. So this is the categorization. All right. So the tegmina, since here it is asking about the tegmina, which is the first pair of wing. All right. So the first pair of wing basically arises from the mesothorax. That means this one, because in case of first prothorax, metathorax, these two are basically responsible for not giving rise to wings. All right. because no wing arises from the prothorax. This is a fact. And then hind wing also, that means from the hind part also of the metathorax, no wings is given rise to. All right. So definitely we can say that mesothorax. So here the tegmina, which is also considered to be as forewing, arises from this part. All right. So that's why the correct answer will be option B over here. While in case of prothorax and metathorax, while no wings arises, prothorax and metathorax. Basically, we can say that no wings arises. That means there is no sign of wings that arises from the prothorax, but if we just look into the metathorax, so metathorax give rise to hind wings. All right, so that is the case. So and metathorax, this give rise to hind wings. All right, so that is the case. So with this, option B is the correct answer over here. Now, let's talk about question number 75. Now, in case of the question number 75, there are two statements that has been given below, and statement 1 and statement 2. So you have to identify the correct statement. That means the most appropriate answer from the options that are given below. All right. Now, statement 1 says that the release of sperms into the seminiferous tubules is called as the spermiation process. All right. And statement first basically says that, and statement 2 says that spermiogenesis is the process of formation of sperms from the spermatogonia. All right. So spermiogenesis basically involves the process of the formation of the sperms from the spermatogonia. All right. So basically, the statement 1, which is saying that the release of the sperms into the seminiferous tubule is called spermiation. So this statement is actually a correct statement. All right. So statement 1 is considered to be correct because yes, spermiation is a process where the sperms are released from the seminiferous tubules after getting nourishment from the Sertoli cells. All right. So spermiation is the process of release of sperms from the seminiferous tubules after getting nourished by Sertoli cells. All right, so this is a correct statement. Now, statement 2, which says that spermiogenesis is the process of formation of sperms from spermatogonia. Now, this is considered to be as an incorrect statement. This is basically wrong and this is right. All right. Why this statement is incorrect? Because spermiogenesis is not the conversion of spermatogonia into sperm, but it is the process of formation of spermatids into sperms. So this is actually incorrect, because spermiogenesis is the process of inversion of spermatids into sperms or spermatozoa. All right, so this is the correct statement actually. So with this, if we just look into the different options, then option C, if you just look into it, that statement 1 is correct, but the statement 2 is incorrect. This is the correct answer over here. All right, now question number 76. Question number 76 says that if 8 Drosophila in a laboratory population of 80 died during a week, the death rate of population is dash individuals per Drosophila per week. All right, so let us just calculate with the given information. Like, given information is that 8 Drosophila, just a second. So 8 Drosophila in a lab population of 80. That means 80 is indicating total population. All right. So they died during a week. All right. So the death rate in the population will be considered to be as the number of deaths that occurs, that is occurring in the population, divided by the total number of individuals that are actually present. All right. So the death rate will be, so the death rate is number of individuals died divided by total number of individuals in the population. So that will basically give an idea of the death rate of the individuals per Drosophila and should be per week. So it is 8 by 80. So this will give rise to 0.1. All right. So this means 0.1 individuals per Drosophila per week is the death rate. All right. So with this, option A is the correct answer. Now, question number 77. So question number 77 says that natural selection where more individuals acquire specific character value other than the mean character value, so that will result into. All right. Now, this is a question that is related to the 3 categories of the natural selection that people might have studied it also. There are 3 types of natural selection actually. It can be stabilizing, it can be directional, and it can be disruptive as well. All right. And this is a very direct question that has been asked. So, because in the statement here, it has been given that more individuals are actually acquiring a particular specific character value than the mean character value. All right. So, that means more individuals are inclined towards a particular direction, not having a central mean character, but inclining towards a particular character or towards a particular specific value. So this is somewhere it is correlatable to directional change. All right. Because in case of stabilizing selection, if we see, so stabilizing selection usually shows a pattern of a mean character value. So there's a mean character value which is always obtained in case of the stabilizing selection, and the mean character value is like this. All right. So definitely this cannot be an option because this is stabilizing selection when more individuals acquire mean character value. So that is the case. So definitely this cannot be the option. All right. Then there is directional change. Directional change, if we will see to it. So directional changes to a particular direction only. We will see to it. Like for example, it's going like this, and there will be a particular. Just a second. So this will go like this, and there will be a particular inclination towards a particular direction. So this is not acquiring the mean character value, but it is going to a particular direction here. So this is directional selection. Where more individuals acquire a value other than mean character value. All right, so that is the case. So this is, you can see this one here is basically the direction is towards one side, like this. Then the C1 is considered to be as the disruptive change. Now, disruptive change is one such kind of a change where the individuals they actually acquire peripheral character value at both the ends of the distribution curve. So if you just look into the graph, so this gives an appearance like this one, which is going to give like this. So they basically acquire, they won't be acquiring the mean character value, but it will look like this. So this is the case. So here, this is disruptive selection or disruptive change, you can rather say. All right, so we can say that more individuals acquires so when more individuals they basically acquires value other than the values that are actually present, or rather you can say this is going to actually acquire. All right, so basically the individuals they will be acquiring the peripheral character value actually. Just a second. So they basically acquires peripheral so they will be acquiring peripheral value at both ends of the distribution curve. At both ends of distribution curve. All right, so that is the case. And the last one is the random change, basically. So random change, again, random change also won't be the option because there is no random changes that happens in case of natural selection. Because if there will be random change, then it won't be it won't be the cause of the natural selection. So definitely random changes, if I say, then random changes does not causes natural selection. So definitely this also won't be an option. All right. So this one is also wrong. And then this one is also wrong. That means option B is the correct answer over here. Now, next is question number 78. Question number 78 says that which of the following is not the function of the conducting part of the respiratory, not the function of the conducting part of respiratory system, it is asking about it. All right. Now, the options that has been given are it clears the inhaled air from the foreign particles. Then option B is inhaled air is humidified. Then temperature of inhaled air is brought to the body temperature and provides surface for diffusion of oxygen and the carbon dioxide. So let me tell you that the conducting part where the conducting part of the respiratory system it is talking about, and it is not, and it is basically specifically asking not the function. So that means you have to find out that which of the following statement doesn't involve the functions of the conducting part. So out of all the functions that has been mentioned over here, so basically option 4 will be the correct answer. Option 4 will be the correct answer, and the reason for this is because if you just look into the start part, so it starts with external nostril. Conducting part, you have to find it out. All right, so definitely the first is going to start with the external nostril. So conducting part of respiratory system. All right, so the conducting part of respiratory system, it starts with external nostrils, and it constitutes up to the bronchus part. That means the bronchioles part basically, and terminates into bronchioles. That is found in the lungs. Into bronchioles found in lungs. All right, so that is the case. While the alveoli that comes after the lungs, that comes after the bronchioles. So alveoli and their ducts, they basically form the respiratory or the gaseous exchange part of the respiratory system, but not the conducting part. Remember it. All right. So you can say that, however, the alveoli and its sacs or ducts, you can say, constitutes the exchange part of the respiratory system. So basically, we can say that these alveoli, that means alveoli basically serves to be as the site for the exchange of the gases between the oxygen and the carbon dioxide. All right. So that means this one. So this function is actually being performed by alveoli. This is not performed by the conducting part of the respiratory system. All right. So what it does is that, so conducting part basically is responsible for inhaling the air. All right. And once the air is inhaled, it clears it from the foreign particles, also humidifies it, and also brings the air to the body temperature. All right. So this basically, we can say that all the three options are correct, but this is not the function. That means D is not the function. So we can say that, we can just write that the conducting part transports the atmospheric air to alveoli, clears it from foreign particles. So it clears it from the foreign particles, humidifies, and also bring the air to body temperature. That means all these three statements are the functions of the conducting part. Only the last one is not the function. So that's why the correct answer is option D over here. All right. Now, question number 78. Now, question number 79. Now, question number 79 says that breeding crops with higher levels of vitamins and minerals or higher proteins and healthier fats is called. All right. So this is basically a definitive direct question that has been asked. Now, the process is considered to be as biofortification. All right. That means option C is the correct answer, because biofortification is a process in which crops are breeded in such a manner that they have high level of vitamins, minerals, and higher proteins and healthier fats. All right. So biofortification. So a method of breeding crops breeding crops with high level of vitamins, then minerals, then proteins, and healthier fats. Healthier fats. All right. Now, regarding the other definitions that has been given, or other terminologies that has been given, so biomagnification and bioaccumulation is completely different thing. Biomagnification is basically the increase in the concentration of the toxicant at the successive trophic level, basically. So biomagnification biomagnification, it is the increase in the concentration of toxicant from, or you can rather say, in successive trophic levels. In successive trophic levels. So definitely this won't be an option. And bioaccumulation is basically the accumulation of heavy metals inside the body of the plant. So that is, or any living tissue, you can say. So that is called bioaccumulation. So it is more or less just like the biomagnification only, but the only difference is that biomagnification is more or less being more profound in case of the aquatic ecosystem. So this is more common in case of aquatic ecosystem, but bioaccumulation is more common in case of terrestrial environment. All right. So these two are basically more or less the same thing. So this cannot be the option. And next is the bioremediation. So bioremediation is actually the phenomena by which a biological organism handles the pollution. That is called bioremediation. So this also won't be an option. So we can say that bioremediation. So a phenomenon of using biological organism to handle pollution. All right. So that is the case. So that means option C is the correct answer. Now, question number 80. Now, in case of the question number 80, it is asked that in the taxonomic categories, which hierarchical arrangement in the ascending order is correct in case of the animals. All right. Now, let us draw the taxonomic hierarchy or the taxonomic categories. So, taxonomic categories. All right. So if we just move on with the descending part, so we are going to go from the in the descending order and ascending order. So I will draw both the ascending and the descending part so you people can have an idea. So if you just move in the descending order first, so it comes with kingdom, then class. This is basically the descending order that I'm talking about. Descending order. All right. So this is then kingdom. Sorry, not class. Just a second. Yeah. So this is the descending order that we are drawing. All right. So this will come with kingdom. Kingdom is followed by phylum oblique division. All right. Then class. Then order. Then comes family. Then genus. And then comes the species. All right. So this is the descending order. So that this indicates that kingdom is the highest category. This is the highest category. And this one, species, is the lowest taxonomic category. All right. Now, if you just draw it in the ascending order, so the ascending order will be opposite. Ascending order. So, in case of the ascending order, We can go like this. So here it will be species, then genus, then family, then order, class, then phylum, oblique division. So phylum is basically used for animals taxonomic category. So if the categorization is for animals, then phylum we use it, and division is for plants. And the last will be kingdom over here. So this is the ascending order. So we have to find out the ascending order. All right. Now let us take a look. So okay. So basically, it's kingdom, then phylum, class, order, family, genus, and then species. Then kingdom, class, phylum, family, order, genus, and species. Then kingdom, order, class, then phylum, then family, genus, and species, then kingdom, order, phylum, class, family, genus, and species. Basically, if you just look into the options, so since this has been, asked about the ascending order over here, so in case of the ascending order, the species should come first. All right, and the genus will be on the kingdom will be on the last category, but here all the options are basically showing the kingdom to be the first highest category. So this is basically arranged in descending form, uh, rather than the ascending order. I guess this question is basically incorrect. The options of this questions are actually incorrect, so none of the options are correct in this category. But if we have to find out in the descending order or in the ascending order, if you will see to it, so in the descending order, this is the correct answer, but this is the correct answer in descending order, remember, not in the ascending order. So with respect to the questions, none of the options are correct. All right, but with respect to descending order, option A is the correct answer. So this is a little controversial question that has been asked, but this is the correct, basically, statement. All right. Question number 81. Now, question number 81 says that at which stage of life the oogenesis process is initiated. All right. So it is asking about the process of initiation of the oogenesis. So this is again a very direct question and an easy question that has been asked. So basically, we all have studied that oogenesis is one such process that begins at the time of embryonic developmental stage. All right, when the oogonia, that means the gamete mother cells, they are formed within each fetal ovary, and once the baby has been delivered, no more oogonia is added or formed after the birth of the human female. So basically option B is the correct answer. So we can just write the explanation that oogenesis is initiated during embryonic developmental stage. So this is basically initiated during embryonic developmental stage when the oogonia are formed within each fetal ovary. All right. So this means oogonia is formed within each fetal ovary. And after the birth of the baby, no more oogonia is added. So after birth of a human female child, female, no more oogonia is added or formed. All right, so that is the case. So this means not at birth, not at the adult stage, not even at puberty, because puberty is at the stage of puberty, the process of spermatogenesis begins. That means the process of sperm formation in case of males. So definitely this won't be an option also. So option B is the correct answer. Now, question number 82. The question number 82 says that you have to identify the asexual reproductive structure that is associated with penicillium. Now, let me tell you that penicillium is basically a category of ascomycetian member of fungi. And the asexual reproductive structures which are found in the penicillium are the conidia. All right. So basically, conidia are the asexual reproductive structures. Are the asexual reproductive structures in penicillium. And this is usually non-motile. This is usually non-motile, like this. All right. So this one will be the case. While zoospore, if you just find out, then zoospores are basically the asexual reproductive spores in algae and in the members of Phycomycetes, basically your Oomycetian members. So zoospores, these are asexual reproductive spores. Reproductive spores in algae. All right. So definitely this will. Gemmules are the asexual reproductive spores in sponges. So gemmules are asexual reproductive spores in sponges. That means members of Sporifera. And again, buds. Buds are the asexual mode of reproduction in case of Hydra. So buds, these are asexual reproductive structures, you can say rather. Structures in Hydra. All right. So definitely option A is the correct answer. Sorry, option B is the correct answer. Now, question number 83. Now, let's talk about question number 83. So, this question says that in gene therapy of adenosine deaminase deficiency, that means ADA deficiency, all right, the patient requires periodic infusion of genetically engineered lymphocytes because, all right, so basically this question has been asked from the chapter of biotechnology and its application of 12th standard, all right, and if you just read out the statements, then the first statement says that retroviral vector is introduced into these lymphocytes, then gene isolated from the marrow cells producing ADA is introduced into cells at embryonic stages. Then lymphocytes from the patient's blood are grown in the culture that is outside the body, and genetically engineered lymphocytes, they are not the immortal cells. All right. Now, this question is basically a question ADA is basically, or this rather, I'll say that this question is basically related to the gene therapy concept. All right. And we know that the first clinical gene therapy was actually given to a 4-year-old girl in the year 1990 with the ADA deficiency, because ADA is such an enzyme that is crucial for the immune system to function. All right. And if a patient is suffering from ADA, all right, because this enzyme, which is ADA, is important for, just a second. ADA is important for immune system to function. Immune system to function. So definitely, if there will be ADA deficiency, then there will be the lack in the immune system of the child. All right. So if you just read the following statements that has been given, so here it has been written periodic infusion. This means that this enzyme should be given regularly to the patient or regularly to the child, all right, in his entire life. So retroviral vector is introduced into these lymphocytes. Yes, retroviral vectors are introduced into these lymphocytes, and which are ADA cDNA. But once these retroviral vectors are introduced into these cells, and we know that basically these lymphocytes, they are not immortal cells. All right. So definitely this cannot be the correct answer, because we can say that the retroviral vector, here it is talking about it, retroviral vector. All right. So retroviral vector here is ADA cDNA. Or rather, we can say that the functional ADA cDNA using a retroviral vector, that would be rather more correct. So a functional ADA cDNA using a retroviral vector. So using a retroviral vector is actually introduced into these lymphocytes. So is introduced into lymphocytes, is introduced into lymphocytes, and once they are introduced into the lymphocytes, they are subsequently returned to the patient and subsequently returned to patient. Subsequently returned to patients. So this cannot be the correct answer, because this method, and since it is here, it is talking about the periodic infusion, that means regular infusion. So that's why this technique, as a first step of this technique cannot be considered to be as a, this will be further converting it into a permanent cure. So for this is actually a process of permanent cure which is actually being given to the patient. So that's why here it won't be the involvement of the periodic infusion. All right. So definitely this cannot be the correct answer. Then the gene isolated from marrow cells, producing ADA is introduced into cells at the embryonic stages. Again, yes, it is actually correct. That means this statement is correct. But so we can say that when the gene will be introduced, or when the ADA will be introduced in the embryonic stages, then that would be a permanent cure. So definitely the periodic infusion of the genetically engineered lymphocytes won't be involved. All right. So again, we can say that gene isolated from marrow cells. That means bone marrow cells, it's talking about it. Producing ADA is introduced into cells at embryonic stage. So if the cells will be introduced during the embryonic stage, so this will be a permanent cure. So this is a permanent cure. Permanent cure. So if this will be a permanent cure, then definitely the periodic infusions won't be involved. So the periodic infusion won't be involved. All right. So that will be the case. So again, this won't be an option. Then lymphocytes from the patient's blood, they are grown in a culture outside the body. Again, this option cannot be considered to be as the correct reason, because as I told you that this is a correct statement with regard to this gene therapy concept, but this is not the correct reason, we can say. So definitely this is also a wrong reason, because C, option C is the correct statement. This is the correct statement with regard to concept of gene therapy. Concept of gene therapy, but not the correct reason. Not the correct reason. So definitely this is also not the correct answer. Now the last statement left is option D, which states that genetically engineered lymphocytes are not immortal cells. Now this is considered to be as the true answer or the correct answer, because we know that the lymphocytes, they have a certain lifespan. All right. And since the lymphocytes, they have certain lifespan, so if the genetically engineered lymphocytes, even if they are introduced into the patient's body, they will require that the patient body will require periodic infusion of these genetically engineered lymphocytes due to limited lifespan, because they will ultimately die after some time, because they are not the immortal cells. So we can say that this is the correct answer. So this is the correct answer, as genetically engineered lymphocytes are not immortal cells, and have limited lifespan. And have limited lifespan, after which they die. After which they die. So definitely the patient will require periodic infusion of such lymphocytes. So option D is the correct answer over here. All right. Now, question number 84. Now, question number 84 says that you have to select the incorrect statement with reference to mitosis process. All right. So this is a question that has been asked from the chapter Cell Cycle and Cell Division of Standard 11th. All right. And the options or the statements that has been given with regard to this question is that all the chromosomes, they lie at the equator at the metaphase. So let us just analyze all these statements. The first statement says that all the chromosomes, they lie at equator at the metaphase state. So basically, this is a correct statement. This is correct because when you just analyze the metaphase stage of an animal cell, so basically it lies at the equatorial plate. So spindle rays like this, it emerges out. Spindle fibers, basically like this. And the chromosomes that you will be seeing, it lies at the equatorial plate. Like this. And these spindle fibers, they are attached like this. All right, so this is the metaphase. You can see. And all the chromosomes, they are aligned at an equator phase. So, chromosomes at equator. Or you can say equatorial plate rather. All right, so this is the true statement. So definitely we have to identify the incorrect statement. So this is not the option. Now the second option says that spindle fibers, they attaches to centromere of the chromosome. Now this is also a correct statement because spindle fibers actually spindle fiber doesn't attaches to themselves at the centromere. So this is actually an incorrect statement, not the correct statement actually. This is actually the incorrect statement because spindle fiber attaches to themselves at a point which is called as kinetochore, not to the centromere. All right. So you can say that spindle fibers attaches to kinetochore of chromosomes. So they basically attaches to the kinetochore that is found in the centromere of the chromosomes. And these kinetochores, these are basically disc-shaped structures. All right. So we can say that kinetochores are disc-shaped structures, that is present on the sides of the primary constriction. So present on the sides of centromere, which is also known as primary constriction also. Centromere of chromosome. All right. So we have found out the incorrect statement. Now let us just draw the figure in order to make you people understand. So if we just draw a sample chromosome like this, for example, and this one is the centromere that you can see. All right. Now, in attachment to the centromere, there is a point here at this. So this point which you can see, disc-shaped structure, it is present on the surface of the centromere, which is called kinetochore. And this one, central one, which you can see over here, this is the centromere. And this is a chromosome. So definitely, this is not the Correct statement. So this is a false statement. So incorrect statement. That means this statement is actually right. But let us take a look at the other statements as well. So other statement says that chromosomes decondenses at the telophase stage. So this is basically true. This is a true statement. That means this is a correct statement. That option C is also correct. Because when you will see at the telophase stage, so in case of the telophase, the chromosomes, they basically lose their identity and they convert themselves in the form of chromatids like this. All right. So they are going to lose their identity like this. So two cells are there. Like this. So chromosomes are decondensed and they decondense to form chromatin. So this is chromatin and this is the telophase. All right. So chromatin formed due to decondensation of chromosomes. Decondensation of chromosomes. So that will be the case. All right. Now, so this is also correct. So that means this won't be the option. Then splitting of centromere occurs at the anaphase. So this is also a correct statement, but since we have to find out the incorrect statement, so this is also not the option. So option D is also correct. So this is also a correct statement because when you just look into or when you just draw the anaphase, so this gives an appearance like this one. For example, this is the asterase that is found in the giving rise to spindle fibers like this. Then basically in case of anaphase, the centromere splits. All right. And when the centromere will split themselves, so this will result into separation of the chromatids, sister chromatids with one another, thereby the sister chromatids, they move to opposite poles. So this is anaphase. And this phase is characterized by centromere splitting, resulting into separation of sister chromatids and their movement to opposite poles. And their movement to opposite poles, as you can see. All right. So that is the case. So with this question, the correct answer is option B over here. All right. Now, question number 85. Now, question number 85 says that detritivores break down detritivores breaking down detritus into the smaller particles. So this process is called as. Now, this is a very direct question that has been asked, from the chapter of your ecosystem, and this is basically from your 12th, standard. And there is a concept of decomposition. Let me tell you before going into the options. So decomposition, you people might have heard it also. And decomposition is basically one such process in which there is conversion, you can say rather, or you can say the conversion of the complex dead organic matter into simple absorbable forms. So we can say, or we can define decomposition as it is a process of conversion of dead organic matter into simple absorbable forms. Absorbable forms. All right, so this is the definition of decomposition. Now, this entire process of decomposition is carried out by two organisms. One is the decomposer and the other is the detritivore. All right, so this is primarily carried out by two organisms. So one is called as the detritivore. Detritivores. So here it has been asking about this process, detritivores. All right, and the other is called as the decomposers. So there is a difference between detritivores and decomposer. They are not same, remember. All right, so detritivores, the example basically includes earthworm. All right, and they are basically the agents or the organisms which are responsible for converting the detritus into smaller particles. So these convert detritus. Now, what is detritus? Detritus is basically the raw material of decomposition, remember. So these convert detritus into small fragments or smaller particles, you can say. And the decomposers, which basically includes microorganisms like bacteria and fungi. So this is going to include the main organisms, which is mainly going to carry out the process of decomposition. So this includes bacteria and fungi. So they convert small fragments. Now they will be converting ultimately the smaller fragments into absorbable forms or into simple forms, you can say, into simplest forms. Simplest forms. So since here it is asking about the different process. So is it catabolism? Is it fragmentation? Is it humification or decomposition? Now let me tell you that decomposition, the entire process of decomposition is primarily divided into four distinct stages. So decomposition is completed in 4 steps. So this is primarily completed in 4 steps. So the first one is fragmentation. So the first one is the fragmentation process. Now fragmentation process is performed by the detritivores. So this means this is a process in which detritivores converts detritus, which is the raw material, complex organic matter, into small fragments. Small fragments or smaller particles, you can say. All right. So that means the statement that has been given over here, with the statement, we can see that option B is the correct answer over here. All right. Definitely the process of decomposition cannot be the correct answer. Option D cannot be the correct answer, because decomposition is a wholesome process which involves fragmentation. All right. So definitely decomposition won't be the case. Then the next two options are catabolism and humification. Let us take a look. Then the first step is basically fragmentation. Then the second one is leaching. Leaching is basically a process in which all the water soluble inorganic solutes, they will be penetrating deep inside the soil. So all water soluble inorganic solutes solutes, penetrates deep down the soil. So definitely this also won't be the option because this is not given in the option actually. Then the third is catabolism. Catabolism is basically a process in which the smaller particles that has been fragmented by the detritivores, now they will be acted upon by the decomposers. So a process of converting small particles into simplest form, you can say, rather, or simple forms, you can say, by decomposers. That means bacteria and fungi. And the last turns out to be, so we can say that definitely here catabolism is also not an option. All right. And the last turns out to be humification. So the last is humification. Humification is basically the process of the formation of humus, which is a dark black colored colloidal substance, rich colloidal substance, and it is nutrient reserve filled, which is enriched with nutrients. So process of humus formation. Humus formation. So definitely this also won't be an option. So that means our correct answer is option number B over here. All right. Now, question number 86. Now, question number 86 says that if a colorblind female marries a man whose mother was also colorblind, then what are the chances of her progeny having the color blindness? All right. Now, in this question, this is a question which is being asked, from the chapter of Principles of Inheritance and Variation of your Standard 12. Now, for analyzing this question, we need to draw a pedigree chart. So we will be drawing a pedigree chart first, and then we will be knowing the fact that what will be the chances. Now let us just draw it. So pedigree analysis. All right. So if you're going to analyze the pedigree of the given question, so it says that the female is colorblind. So if the female is colorblind, so definitely she is the affected one. All right. So this is the colorblind female. All right. Now this colorblind female is going to marry a man whose mother was also colorblind. All right. So we can say that the male will also be colorblind. So definitely we can say that the male will also be colorblind. All right. So if the mother was also colorblind, that means this person's mother. All right. This male's mother was also colorblind. So this means the chances are this one. All right. So definitely one of the that means this mother is basically the affected mother. So that definitely one of the chromosomes, carrier chromosomes, will be transferred to the male also. So that's why we have written the probability over here that the male will also be colorblind, because the colorblind gene is carried on X chromosome, and males have one X chromosome. So definitely they will be carrying it. So this is colorblind male. This is colorblind male. All right. So that is the case. Now, what are the chances of her progeny having the color blindness? Now, let us draw the Punnett square with the help of this. So, if we just analyze the Punnett square, so we can like draw like this, giving a pictorial representation. This one, then like this. So at one side we can draw the female, and on the other side we can draw the male like this. If you just draw it, X, the combinations. So this is going to be like this, then this, and then this. So it is asking that what are the chances of a progeny having the color blindness. So if you will just analyze the probability, then all the progeny is having color blindness. So this is also color blind. All right. This is also color blind, CB. Then this is also color blind. That means two female babies are colorblind and two male babies are also colorblind. So that means wholesome, we can say that all the progenies are colorblind. All progenies are colorblind. So the chances of their baby to be colorblind is 100%, we can say. 100% chance is there. So out of all the options, option D is the correct answer over here. All right. And in this process, and this let me tell you that if the mother of a man is considered to be colorblind, as I already told you that the mother of a man is considered to be colorblind, then due to crisscross inheritance, all right, due to the process of the crisscross inheritance, then the son of that mother will also be considered to be colorblind, because this is a X-linked recessive trait, and that follows the pattern of crisscross inheritance. That means the trait will be transferred from mother to son and son to daughter. All right. So let me tell you this also, that color blindness is a X-linked recessive inheritance. So this is an X-linked recessive inheritance that follows the pattern of crisscross, or we can say that follows crisscross inheritance pattern. Crisscross inheritance pattern. All right. That is, we can say trait is transferred from mother to son and son to daughter. So this is considered to be a crisscross inheritance. All right. So if the mother is affected, then definitely the male child will be affected. So since here the mother is affected, so the male child will also be affected. So option D is the correct answer. Now, question number 87. Now the question number 87 says that you have to select the incorrect statement with respect to acquired immunity. All right. Now this, remember, you have to select the incorrect statement. Now this question has been asked from the chapter of human health and diseases of your standard 12. And we have read out some of the important features of the acquired immunity, because acquired immunity, we all know, is an immunity which we acquire during the lifetime, but not from the birth. All right. So basically, we can say that acquired immunity is a type of immunity or defense, you can say, that is acquired during lifetime, but not from the birth. Acquired during lifetime, but not from birth, or not at the time of birth, actually. All right. So let us take a look into the other options, actually. What are the different options that has been given? So since you have to find out the incorrect statement. So the first statement says that the primary response is produced when our body encounters a pathogen for the very first time. All right. So definitely, this is a correct statement. Option A is correct. This is correct because, as I told you, that when our body will be encountering the pathogen for the very first time, so the first response that the body will be producing, acquired immunity basically produces two types of responses. One is the primary response, and the other is the secondary, which is also known as amnestic response. All right. So, primary response will be produced in a body when the body will be encountering the pathogen for the very first time. So, this is a correct statement because when our body encounters pathogen for the first time, it produces primary response or primary immune response. Primary immune response. So this is actually a correct statement. All right. Now, the option B says that amnestic response is elicited on subsequent encounters with the same pathogen. Now, let me tell you that here amnestic response basically means secondary response. All right. And this is also a correct statement. This is a correct statement, remember. This is considered to be as a correct statement, not the incorrect statement, basically. So we can like remove this part. So this is a correct statement, and this is also a correct statement. Now, why this is correct? Because secondary, as I told you, that acquired immunity is producing two types of responses, primary and secondary. Now, secondary response will be produced once a pathogen will be encountering the body, anytime during the lifetime. All right. And on subsequent encountering, you can say, with the same pathogen. So, it's just a moment. So, an amnestic response, which is also considered to be as secondary response. So, anamnestic or secondary response is activated due to subsequent encounter with same pathogen. So, due to subsequent encounter with same pathogen. All right. So, this is also correct. Now, the option C is amnestic response is due to memory of first encounter. Definitely. So, this is also considered to be as a correct statement that this secondary response is such a highly intensified response that encounters the memory of the first pathogen. So, we can say that secondary, or rather, we can say amnestic. An amnestic response is a highly intensified response that encounters the memory, or you can rather say that it basically encounters the memory. It is due to memory rather, not, we can rather say not encounters statement little. Okay, so anamnestic response is a highly intensified response that is due to memory, we can say. That is due to memory of the same pathogen when the body encounters with the same pathogen. So this is also a correct statement. Now the last statement is only left, which is acquired immunity is a non-specific type of defense present at the time of birth. No, this is considered to be incorrect because the very first important definition that I told you, that means this one is only that acquired immunity is a type of immunity or defense that is acquired during lifetime but not from the birth. So definitely this is an incorrect statement. All right. So option D is the correct answer. Now, question number 88. Now, question number 88 says that statements related to human insulin, they are given below. Okay. Now, which statement is or are correct about the genetically engineered insulin? So, this question has been asked from the chapter of biotechnology and its application. And this is regarding the genetically engineered insulin of your 12th chapter, basically, of your 12th syllabus. So, if we just look into the different options that has been given, so we have to find out the correct one. All right. So if we just draw the, insulin. So let me tell you that the first statement which says here is that pro-insulin, pro-hormone insulin contains an extra stretch of C-peptide. So this is basically correct. This is considered to be as a correct statement because if you just draw the pro-insulin, so they have an additional stretch of C-peptide. And that's why they are actually inactive. This is something like this. And here there is again an extra stretch over here. So this is pro-insulin. All right. Then these are basically attached with the help of disulfide bonds like this. So it's the disulfide bond that attaches themselves because this is a protein molecule, right? So that's why they are attached to it. And this extra stretch it is talking about, this is the C-peptide actually. This is the C-peptide over here. We can like fulfill, fill this box in order to make a differentiation between the A peptide and the B peptide actually. All right. So basically this one you can see, this one is the A peptide. This one I'm talking about. So this is the A peptide over here. All right. And this one is considered to be as the B peptide over here. Like this. And once the elimination, how the process goes on is like that, because since C peptides is an extra stretch, so this peptide is basically an extra stretch. So extra stretch. So in order to make this insulin active, we need to remove this extra stretch. That means removal of C peptide. Removal of C peptide. Once the C peptide will be removed, it results into insulin formation, active insulin formation. Insulin formation from proinsulin, basically, which is inactive. And now the insulin will be active. So how it goes like this, here also an additional stretch is found, C-peptide like this, disulfide bond. So we can like remove This will be the A peptide. We can like fill like this. Then plus the B peptide will be like this. All right. And they will be ultimately joined with the help of, you know, disulfide bonds like this. All right. So this is A, this is B peptide, A peptide, and this is B peptide, and plus along with that, there will be a free C peptide. There won't be the free peptide is now, that means the free peptide is now the C peptide, this one. So this is free C peptide. All right. And now this too, just a second. This too now forms the insulin. So insulin is basically a combination of A and B peptide actually. All right, so this is how the process takes place. Now let us take a look into the other options. So this is a correct statement. Then the B statement says that A peptide and B peptide chains of insulin, they were produced separately from the E. coli, extracted and combined by creating disulfide bond between them. Now, this statement is also, if you just consider it, this statement, then this statement is considered to be as actually true. So basically this statement, that means option B over here, this is also considered to be as a correct statement. All right, so this is also correct. As you can see that A and B peptide chains that are found in this proinsulin, this one is the A peptide, then B, this one is the B peptide, but in the insulin, that means in the mature insulin, if you can see, it is basically linked together by the disulfide bridge. So this is the disulfide bond which is connecting the A peptide with the B peptide. So this is also a correct statement. Now the option C says that insulin which is used for treating diabetes was extracted from cattle and pigs. All right. Now this statement is basically the incorrect statement. This is actually incorrect because it was extracted. That means earlier the insulin for treating the diabetic patients, it was extracted from the slaughtered pancreas, from the pancreas of the slaughtered cattle and pigs. All right, so we can say with the C option, if we just write it, then earlier you can say, earlier for treating the patients, for treating diabetic patients, insulin was extracted from the pancreas of slaughtered cattle and pigs. Remember, it is slaughtered cattle and pigs, not the cattle and pigs which are living. All right, and that too from their pancreas. So definitely this is an incorrect statement. Now, the next statement is that pro-hormone insulin needs to be processed for converting into a mature and a functional hormone. So now you can see this statement is also considered to be as a correct statement because in the given figure below, you can see that we have converted this pro-hormone insulin for getting, and this is basically inactive. This pro-insulin is considered to be here as inactive. So for converting this inactive pro-insulin into an active insulin, the processing has to be done. So this entire process is called as processing. So this entire thing which you can see over here, this entire process is processing only. So the entire processing is done, and then only the proinsulin gets converted into insulin. So that's why we can say that this is a correct answer. statement. All right, and the last statement is that some patients, they develop allergic reactions to foreign insulin. So this statement is considered to be as an incorrect statement, because if the insulin is extracted from an animal source, all right, so if the insulin is extracted from an animal source, then it could be the development of allergic reactions. But in this statement, it is not specifically mentioned that whether it is talking about the genetically engineered insulin, that means insertion of the genetically engineered insulin into the diabetic patient, or the insulin that has been extracted from the animal source. So definitely we cannot consider the statement to be as the correct one. All right, so we can say that E statement, basically for the E statement, we can say that if the insulin is extracted from animal source, if insulin is extracted from an animal source, then it could be sorry, then it could have allergic reactions to the patients. Then it could show allergies to patients. Remember. So definitely this cannot be the correct statement. So with regard to this, if we just look into the options, then option A, B, and D. A, B, and D is considered to be as the correct answer. All right, that means option number A is the correct answer. Now, question number 89. Question number 89 says that which one of the following statement is considered to be as the correct statement. All right, now this question, which you can see, now let us first take a look into the following statement. Have a look. So the first statement says that the atrioventricular node, that means AVN, generates an action potential to stimulate the atrial contraction. Then the tricuspid and bicuspid valves open due to pressure exerted by the simultaneous contraction of the atria. Blood moves freely from atrium to the ventricle during the joint diastole, and increased ventricular pressure causes closing of the semilunar valves. Now, this question has been asked from the chapter of your circulation, that means blood circulation of your standard 11th, and this is particularly being asked from the cardiac cycle. All right, now let us just understand the different options that has been given. Let us just talk about the first statement. So the first statement says that the AVN generates an action potential to stimulate the atrial contraction. This is a complete incorrect statement, because the action potential is not generated by AVN. It is the SAN, which is the sinoatrial node, that generates an action potential, and that's why it is considered to be as the pacemaker of the heart. All right, so it is the sinoatrial node, which is shortly termed as SAN, that generates an action potential. It's potential. So that generates an action potential, and that's why called as pacemaker of heart. Called pacemaker of heart. So definitely this is a wrong statement. That means the incorrect statement. Now, option number B says that the tricuspid and the bicuspid valves opens due to the pressure exerted by the simultaneous contraction of the atria. All right. So let me tell you that this statement is also considered to be as incorrect. So this is also incorrect because the opening of the bicuspid and tricuspid valves is because of the pressure that is exerted by the blood that is present in the atria. It is not because of the contractions of the atria, simultaneous contraction of the atria, but because of the pressure that is exerted by the entering blood into the valves or into the atria, you can say. And then there is a decrease in the pressure in the ventricles during the ventricular diastole process. All right, so we can say that B, in case of B, the tricuspid and bicuspid valves opens due to pressure exerted by blood. So it is due to the pressure that is exerted by the blood present in the atria. All right. So that is the case. So this is also incorrect. Now the next statement is that blood moves freely from atrium to the ventricle during the joint diastole. So this statement is considered to be, that means option C is considered to be as actually an correct statement, because let me tell you that joint diastole is one such stage. This is the very first important state of the cardiac cycle, where all the four chambers of the heart are in a relaxed state and there is filling of the blood in the atrium. All right, so we can say that during joint diastole, all four chambers of heart are in relaxed state. So all the four chambers of heart are in a relaxed state and the blood starts filling up in the first it will be start filling up in the atrial chambers, and the blood starts filling in atria, which ultimately results into opening. This creates the pressure and this will result into opening of the bicuspid and the tricuspid valve, and the blood therefore starts filling in the atria, followed by opening of tricuspid and bicuspid valves. And when these valves will open, so definitely the blood will move from the atrium to the ventricles. And so the blood moves from atrium to ventricle. Moves from atria to ventricles. So this is also considered to be as ventricular filling, which is called ventricular filling. So this is a correct statement actually. Now the last statement, which says that increased ventricular pressure causes closing of the semilunar valve. So this is considered to be as incorrect statement because it is not because of the increased ventricular pressure, it is because of the decrease in the ventricular pressure that results into closing of the semilunar valve and thereby producing the second heartbeat sound, which is called dub. So option D. So this is incorrect because closure of semilunar valves that produces second heartbeat sound, which is called dub, is due to decrease in ventricular pressure. All right, so this is also a incorrect statement. So the correct statement is option C over here. Now, question number 90. Now, question number 90 says that this is basically a match the column kind of a question that has been given over here. All right, and you have to list the A part with that of the B molecule. So let us just quickly do it. And this is basically on one side, the different kinds of molecules has been given. So this is basically a question that has been asked. This match the column question has been asked from your biomolecules chapter of standard 11th. All right, now let us take a look at what are the different options that are available. So the first option says it's for glycogen. Now let me tell you, glycogen is basically a storage product. This is the reserve food material or stored product in animals. So this is stored product in animals. So since this is a stored product in animals, so definitely this is going to match up with a fourth one. All right. Then the globulin, the second is globulin. Globulin is basically a globular protein actually. All right. And this globulin is an example of antibody. All right. So globulin. All right. So globulin is a category of antibody because globulins basically means immunoglobulins. All right. And the globulins which forms antibodies, they are considered to be as immunoglobulins. So globulins forms antibodies, you can say. Particularly, you can say immunoglobulins. Immunoglobulins. All right. So that means B is going to match up with here, option 3. Then steroids. Steroids are basically one of the categories of hormones. Hormones can either be proteinaceous or hormones can either be lipid soluble, which is also considered to be as steroidal forms. Like steroid hormones basically includes testosterone. All right. Then estrogen. These are all the steroidal categories of hormones. So again, steroids are hormones. They basically form hormones like testosterone, estrogen, like these such hormones. So C is going to match up with first. All right, and the last is thrombin. So this is going to match up with D only, this biocatalyst. So thrombin is considered to be as a biocatalyst because it is responsible for converting fibrinogen into insoluble fibrin. And so it is responsible for forming blood clot. All right, so thrombin is a biocatalyst. So a biocatalyst converting soluble fibrinogen to insoluble fibrin. All right, which forms blood clot, or which results into rather blood clot. Blood clot. Clear? That is the case. So this means this is going to match up with D. So let us choose the options given below. So A will be matching up with 4, this one. Then B is going to match up with 3rd, that means this one. Then C is going to match up with 1st, and D is going to match up with 2nd. So that means option D is the correct answer over here. All right. Now, question number 91. Now, question number 91 says that 10 E. coli cells with nitrogen content, that means heavy nitrogen content, of double-stranded DNA, they are incubated in the medium containing lighter nitrogen molecule nucleotide. All right. Now, after 60 minutes, how many E. coli cells will have DNA totally free from the heavy nitrogen? So let me tell you that this question is basically, this is a question that has been asked from the chapter of molecular basis of inheritance of your chapter 12. All right, and this is an experimentation question that has been asked. Messelson and Stahl's experiment, who performed an experiment on the heavy and light nitrogen isotopes, or isotopes of nitrogen. So here is basically N15 represents, or rather we can write like this. So this is heavy nitrogen, and this is considered to be as light nitrogen. So these are basically, there are 2 isotopes of nitrogen, heavy and light. All right, now let us just perform the experiment in order to understand it. So the first statement which is saying is that, let us just talk about the first generation. So if we just take a look into the first generation, then it is saying that 10 E. coli cells are having heavy nitrogen of double-stranded DNA, and they are incubated in a medium containing the light nitrogen. So initially it is heavy nitrogen containing 10 E. coli cells. Yes, E. coli cells. Yes. 10 E. coli cells. All right. So we know that the generation capacity of E. coli cells is every 20 minutes. That means they are going to produce themselves. They are going to undergo the process of reproduction in every 20 minutes. So in every 20 minutes, if they will be doing it, so definitely they will be producing 20 cells. So 10 cells will be converting into 20 cells. In 20 minutes, 10 cells will be converting into 20 cells. And as here it has been written that 10 E. coli cells, the incubation has been initially done in a medium containing the light nitrogen as well. All right. So 20 cells are there, and all the 20 cells will be ultimately hybrid, because there a combination of light nitrogen has also been put into. All right. So all hybrid will be there. We can write like this also, like containing A along with the light nitrogen incubation, or you can say incubated with light isotope of nitrogen is also there. All right. So that's why all hybrid cells will be there over here in every 20 cells. So we can write like this. We can write like this. All right. So all will be hybrid. Now, if we just produce the second generation, now let us just talk about the second generation. Second generation will be of 40 minutes. All right. So these 20 cells, which are all hybrid, that is N15 and N14 over here. Now, again, they will undergo the process again in 20 minutes. So 20 and 20 will make up for 40 minutes. All right. Now, these 20 cells will be further producing 40 cells. They will be giving rise to 40 cells. Now, out of these 40 cells, the 20 cells here will be having hybrid. So 20 cells are produced with hybrid. All right. And 20 cells with only light isotopes. 20 cells with this one. Here we can write like this. All right. So that is the case. Now let us take a look into the third generation. Now that means the last generation, because in the question it has been asked that after 60 minutes, how many E. coli cells will be there to have the total will be having the DNA which is totally free from the heavy nitrogen isotope. So the third cell will be the third generation will be the final generation, which will count for 60 minutes. So in case of the third generation, the 40 cells that has been produced over here, all right, which are 20 cells hybrid and 20 light isotope of nitrogen. So 20 cells with this and 20 cells with this. That is the case. All right. So again, they will undergo the process of replication. And when they will replicate themselves, so they will be further producing 3 types of combinations. That means 20 and 14 cells, then 20 hybrid cells will be produced. That means combination of both of them, both isotope, and apart from that, 40 and 14 cells will be produced like this. All right. So let us take a look that what are the options that has been given. So if you just take a look into the options, so total cells that will be produced, that will be left with, that will be free from N15 will be considered to be as, you can see over here, this one and this one. That means 60 cells. 60 cells of this one. Clear? So that is the case. So that means option C is the correct answer. Now, question number 92. So question number 92 says that the recombination frequency between the genes A and C is 5%, B and C is 15%, B and D is 9%, A and B is 20%, C and D is 24%, and A and D is 29%. Now, what will be the sequence of these genes on a linear chromosome? Now, this question has been asked from the chapter of basically principle of inheritance and variation of your standard 12. And this is a chapter, this is a question that has been asked from the linkage and recombination part. All right. So for this, we just need to know we just need to arrange the sequences and that too in order. All right. So let me first tell you that what are the given facts that has been given. So A and C. Basically, these are the recombination frequency. So recombination frequency. All right. So recombination frequency between A and C is 5%. Then it's B and C is 15%. Then B and D is 9%. Then A and B is 20%. Then C and D is 24%. Then it's A and D is 29%. All right. So that means 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay. So the given data has been there. All right. So let us know some of the facts. The very first important fact is that 1% of recombination frequency basically means 1 centimole. All right. So 1% recombination frequency basically means 1 centimole, cm. All right. And in order to place these genes on a linear chromosome, we need to have a decreasing order of the recombination frequency. All right. So for placing genes on a linear chromosome, for placing genes on a linear chromosome, we need to follow decreasing order. So we need to follow decreasing order of recombination frequency, RF. All right, that is the case. So what can be considered? So let us just draw it. This parameters. So if you're going to draw this, then so the first is going to be the decreasing order basically means that we have to start it with the highest number of the recombination frequency, and the highest number here is basically represented bit of A and D. This is the highest. This one is the highest one with the recombination frequency over here. So we'll first draw it like A here, and then we can do it like this. A here and D here. All right. So this basically means that all the genes will be lying between A to D, because this is the highest frequency that we are doing it. So this is 29%. A to D is 29% over here. All right. Now, after 29%, it is 24%. All right. And the 24% is between C and D, as you can see. So that means basically you can say that C and D, since C and D will be covering the major distance. So just a second, we can just write a little closer here. Here will be D, and we can write 29% over here, both of them. All right. Now C will be somewhere over here. All right, because C and D, they are covering the major percentage. They are covering the major percentage, and it is between 24. So here to here is basically 24%. All right, so that is also covered up. Now the next is 20%, which is between A and B. So definitely it will be between A to B, and the distance will be here. A to, or rather we can say here, A to B, which will be considered to be as 20%. A to B, so this will be 20%. All right. So this has been done, this has been done, and this has been done. Now, the next is B to C, which is 15%. So year to year, if you just calculate it, all right. So year to year, it is 15%. It is 15% over here. All right. Then B to D is 9%. So B to D is 9%. So this can be 9% over here. All right. And then the last one, which is left, is A to C, which is 5%. So from year to year, it is only 5%, like this. So that means the sequence will be A, C, B, and D. All right. So the sequence will be A, C, B, and D. So sequence of gene is A, C, B, D. All right. That means option D is the correct answer. All right. Now, question number 93. Now, question number 93 says that which of the following are not the effects of the parathyroid hormone? Now, this is basically asking, this question is a question from your 11th standard and 11th syllabus, and this is from the chemical control and coordination chapter. All right. And here certain functions of the parathyroid hormone has been given, and you have to find out that which is actually not the effect of the parathyroid hormone. All right. So let us quickly go step by step. That means option A over here. So option A basically says that it stimulates the process of bone resorption. So let me tell you about the features of the parathyroid hormone. So basically, the first important feature of the parathyroid hormone is that this is a hormone which is considered to be as a hypercalcemic hormone. That means it increases the blood calcium level. So basically, PTH. PTH is basically the short form of the parathyroid hormone. So PTH is a hypercalcemic hormone. So this is a since this is a hypercalcemic hormone, so it will be increasing the blood calcium level. So it increases blood calcium level. It increases blood calcium levels. All right, so this is going to increase the blood calcium level. Now, the next important point to be noted here, it has been written basically that, okay, so here the first statement that has been given over here is that it stimulates the process of bone resorption. So definitely parathyroid hormone is a hormone that is going to, since it increases the blood calcium level, so definitely this increase in the blood calcium level increases the calcium of increases the absorption of the calcium from the digested food, and definitely there will be more amount of calcium that will be absorbed by the bones. So this increases the blood calcium level, and again this stimulates bone resorption. So this is an effect of the parathyroid hormone. So this cannot be the correct one. It is asking here not the effects, remember, but this is an effect actually. Then the next option says that it decreases the calcium level in the blood. Again, this is not the effect. This is not effect of PTH, because it is not going to decrease, rather it is going to increase the blood calcium level. All right. So definitely this is a correct statement with regard to the question that has been asked. Now, resorption of calcium by the renal tubules, yes. This parathyroid hormone is also responsible for reabsorption of calcium by the renal tubules, actually, because as I told you that it will be increasing the absorption of calcium from the digested foods. So definitely this is also a function. This is also one of the function of the PTH. So we can say also a function of PTH. Clear? Now decreases the absorption of calcium from the digested food. Again, it doesn't decreases, it rather increases. So PTH increases the absorption of calcium from digested food. Absorption of calcium from digested food. All right. So that is the case. So this means B option is not the effect. Then this D option is also not the effect. This also comes under not the effect option. All right. Then the last is it increases the metabolism of carbohydrates. All right. No, it has nothing to do with the basically the carbohydrate metabolism, because the hormone which is responsible for regulating the carbohydrate metabolism are the glucocorticoids and not the PTH hormone. So definitely this is also not the effect. Not the effect, because we can write here rather the statement that carbohydrate metabolism is regulated by glucocorticoids and not by PTH. All right, so this is also not there. So with this, option D, then option D and option E, they are considered to be as the correct one. Option B is the correct answer. All right, now question number 94. Now, question number 94 says that which of the following is not a desirable feature of a cloning vector? All right. Now, which of the following is not considered to be as a desirable feature of cloning vector? Now, let me tell you that this question has been asked from your biotechnology principles and processes chapter of your standard 12, and not a desirable feature, we have to go for it. So let us take a look into the features that has been given. There is presence of ORI, which is origin of replication, presence of a marker gene, presence of single restriction enzyme site, and presence of two or more recognition site. Now let me tell you that cloning vectors are basically considered to be as the carriers, or they are basically the vehicles that are used to carry a desired gene into a particular host cell. So they have certain prerequisites in them, all right, that are found in them. So let us take a look at what are the prerequisites. So desirable features. So cloning vectors are the carriers or vehicles for carrying a desired gene. So for actually carrying a desired gene into the host cell, Now they have some important features. So important features includes the very first important feature, that they have the presence of origin of replication. So presence of ORI, which is considered to be as origin of replication. So this is true. So that means yes, this is, and remember it is asking here not a desirable feature. All right. So there is a presence of origin of replication actually. All right. Not. All right. Then the second important point is that they have the presence of a marker gene. Definitely they have presence of marker genes. Marker genes. All right. Then they have the presence of single restriction enzyme sites, which are basically the antibiotic resistance gene sites. So yes, there is a presence of the, few, that may be single also, or that may be few also basically. So you can say that presence of restriction sites or enzyme restriction sites. So presence of restriction enzyme sites, which are usually the antibiotic resistance gene sites. Enzyme sites. So this is also true. And presence of 2 or more recognition sites. Usually cloning vectors. That means cloning vectors, as I told you, that they are the carriers of vehicles, and the best example of cloning vector is E. coli. So usually when E. coli or any other cloning vector, if we talk about, so they usually have only one recognition site in their entire vehicle or in their entire carrier. They do not have 2 or more. They only have a single recognition site. All right. So presence of single recognition site. There is presence of only single recognition site that is commonly used for restriction enzymes. So commonly used for restriction enzymes. All right. So with this, we can conclude that option D is basically the incorrect answer. That means it is not a desirable feature of a cloning vector. All right. Now, question number 95. Now, question number 95 says that which of the following statement is not true? All right. Now, certain statements are given below with regard to the evolution chapter, and which is of standard 12th, and you have to find it out the not true statement. That means basically the false statement. You have to find it out. You have to find it out the false statement. All right. So the very first important statement says that analogous structures are a result of convergent evolution. Basically, there are two types of structures are found. One that is there in your course, and you people might have studied it also. There are homologous structures, and then there are analogous structures. All right. So homologous structures are the structures that shows divergent evolution, while analogous structures are the structures that shows convergent evolution. Because homologous structures are considered to be, let me tell you, that homologous structures are the structures that have same structure, that will be having the same structures in the different organism, but their function will be different. But that means they are morphologically similar, but have same structure means morphologically similar, you can say. Morphologically similar, but functionally different. But functionally they are different. All right. And since they are morphologically similar, so definitely they will be indicating what? They will be indicating common ancestry. So homology indicates common ancestry. So they indicate common ancestry, and apart from that, they will be ancestry. Sorry, and apart from that, they will be indicating the divergent evolution. So we can write here also, and shows divergent evolution. And they basically shows divergent evolution. All right, like if we just look into the options that has been given over here. So analogous structures, the entire opposite condition is that of the analogous structure. So analogous structures are those structures which are the result of convergent evolution. So true, this is particularly true, because if homology will show divergent evolution, then analogy will show convergent evolution. So this is a true statement. All right. Then apart from that, uh, sweet potato and potato is an example of analogy. Now, sweet potato and potato is considered to be as an example of analogy. So this is also a true statement, because this in case of the sweet potato and in case of the analogy, the morphological structure may be different, but functionally they are similar. So we can say that analogous structures are morphologically different, so they are morphologically different. Sorry, it's different. Morphologically different, but functionally similar. All right. So example is sweet potato and potato. Now, how they are morphologically different and functionally similar? Because morphologically different, they are different because sweet potato is a root modification. So this is a root modification, while on the other hand, potato is an example of stem modification, and this is a stem modification. But how they are functionally similar? They are functionally similar because both of them stores food. But you can say both of them stores food. All right? Stores food. So their functionality is same. So definitely this is also true. Homology indicates common ancestry. Yes, we have seen over here that homology will be indicating common ancestry. So this is also true. Then flippers of penguins and dolphins are a pair of flippers of penguins and dolphins are a pair of considered to be as a homologous organs. Now this is considered to be as a false statement. That means this is not the true statement, because flippers of penguins and dolphins, they are the examples of analogous organs in case of the animals. That means they are morphologically different, but they will be performing the same function, and that too it helps in swimming. All right, so here we can say both structures are analogous. Are analogous as they are morphologically different, but performs function of swimming. Function of swimming. That means their function is same. All right, so that's why the correct answer will be option D over here. Yeah. Now, question number 96. Now, question number 96 says that you have to select the incorrect statement regarding the synapses. All right. So basically, 4 statement has been given, and this question has been asked from your 11th. This is basically a question of your 11th syllabus. And, all right. And that too, it has been asked from the chapter of your locomotion and movement. All right, and let us just take a look that what are the different conditions that has been given with regard to the chemical impulse transmission, or you can say the synapses. All right, so the membrane of the presynaptic and the postsynaptic neuron are in close proximity in an electrical impulse. All right, so remember, you have to select the incorrect statement. All right, so the first statement which says that the membrane which carries the this one, that means the presynaptic and the postsynaptic neurons, that will be in close proximity to one another. So definitely this is a true statement. So this is actually a correct statement. This statement is exactly similar. All right, so this is, we can say this is a correct statement, because the presynaptic membrane and the postsynaptic membrane, they are supposed to be in closure proximity. So this is correct. Now, the next statement says that the electrical current can flow directly. Basically, if I just draw it, let me just draw. So this will somehow give an appearance like this. Like this. So presynaptic is basically, this is basically the presynaptic neuron. All right. And of course, in correlation to this, the postsynaptic neuron is in close proximity, which is basically the dendrite. So presynaptic neuron is the end of the axon terminal of one neuron. So this is basically the axon terminal of a neuron. Axon terminal of neuron. All right. While this is the dendrite, this is the postsynaptic neuron. And the chemicals are released over here in the form of vesicles like this. They are filled up in the vesicles like this, and they come in contact to here, this membrane like this. All right, like this. So this is postsynaptic neuron. So definitely they are in closure proximity. Close proximity. All right, now electrical current can flow directly from one neuron into the other across the electrical impulses. Yes, definitely it can directly occur through it, because in case of the electrical synapses, the transmission of the impulses occurs in the form of electric current, and that electric current will flow from one neuron to the another neuron. So this is also a, basically there are two types of synapses. All right, electrical and chemical synapse. So in case of the electrical synapses, the electric flow occurs between two neurons, and in case of chemical synapses, there will be the chemicals which are being released. All right, so this is also true. Then the next is that chemical synapses uses neurotransmitters. Again, this is also considered to be as a correct statement. This is also considered to be as a correct statement, because here, which you can see here, as you can see over here, this is basically the neurotransmitters only, which are released in the form of chemical between the presynaptic neuron and the postsynaptic neuron. And these are the chemicals only. Chemicals. All right. So that's why the chemical synapses will be using. This is a true statement. Now the last is that impulse transmission across a chemical synapse is always faster than across the electrical synapse. This is a complete incorrect statement because we all know that the flow of electricity is always higher. All right, and since the electric flow will be always higher between the two neurons, so definitely the electrical synapses are considered to be more faster as compared to chemical synapses. So that's why this is considered to be as an incorrect statement. All right, so here we can say that electrical synapses are more faster. Electrical synapse is more faster than the chemical synapse. All right, so that means option D is the incorrect answer. All right, now question number 97. Now question number 97 says that this is basically a match the column kind of a question, and this has been asked from your animal tissue type. All right, and you have to match the column with regard to the different options that has been given below. All right, so let us take a look. Option A, which is bronchioles. Bronchioles. All right. Now, bronchioles, they basically have the presence of ciliated epithelium because anything, and this ciliated epithelium has basically the presence of fine hair-like projections, which is called cilia. All right, and it basically functions in moving the particles of the mucus in a specific direction over the epithelium. So we can say that this is with A option. So bronchioles have ciliated epithelium. So they usually have ciliated epithelium, which is presence of cilia. Presence of fine hair-like projections on its inner surface. So it is the presence of fine hair-like projections. Sorry, this is called actually cilia, which is cilia on its inner surface, which helps in moving the particles, which helps in moving particles or mucus in a specific direction. All right, so that is the case. Now, option B says it's talking about the goblet cells. Now, goblet cells, they have the presence of glandular tissue. Now, let me tell you, goblet cells are basically the unicellular glands. These are a kind of a gland category, goblet cells. All right. So, these are unicellular glands actually. Unicellular glands specialized for secretion. Specialized for secretion. All right. So definitely this is a glandular tissue. So again, this is with B option. All right. Then the third is tendons. Tendons are basically a category of dense regular connective tissue. This is a connective tissue basically, and that it's a dense regular connective tissue. All right. So this is going to match up with C over here. And the last is adipose tissue, which is a category of loose connective tissue. So we can just write first that tendons, tendons basically joins muscles to bones. So are dense, regular, connective tissue joining. It's joining muscles to bones. All right. While the areolar tissue, sorry, it's adipose tissue, I guess. Yeah, it's adipose tissue. Adipose tissue. This is basically a category of loose type of tissue. So a type of loose connective tissue. So it's a type of loose connective tissue that is usually present beneath the skin and stores fat. So present beneath skin and storing fat. All right, now let us take a look into the different options. So A is going to match up with 4, B is going to match up with 3, then C is going to match up with 1, and D is going to match up with 2. That means the correct answer is option A over here. All right, now the next is question number 98. Now question number 98 again is a match the column kind of a question that has been asked, and you have to list the one with that of the two with respect to the methods of the contraception and their respective actions. All right. So this question has been asked from the chapter of reproductive health from your 12th standard. So let us take a look into the different categories. So the first category it is asking about is the diaphragms. All right. So diaphragm is basically a category of it's a category of physical barrier in which is used in case of females. All right, and this is usually done to cover the cervix for blocking the entry of sperms. During the sexual intercourse. So you can say barrier method of contraception. This is basically a barrier method of contraception in females, or you can say for females. For females, there it covers cervix. So this is going to cover the cervix for blocking the entry of sperm. Sperms. All right, so that means this is going to match up with A is going to match up with fourth option. All right, now the next is the contraceptive pill. Now contraceptive pills are such kind of pills which plays a very important role in inhibiting the process of ovulation also and implantation also. All right, because these contraceptive pills, they are made of hormonal preparations, which is usually either progestogens or either progestogen and estrogen combination, and which basically targets these two processes. All right, so we can say that the B is contraceptive pills. So contraceptive pills are made of progestogens or progestogens estrogens combination. Combination that inhibits ovulation and implantation. All right, so this is the case. So this is with B. Now the next is intrauterine devices. Now intrauterine devices, which are also considered to be as IUDs. Now IUDs play a very important role in the phagocytosis process. This is the key word for IUD, which is phagocytosis. So it basically increases the phagocytosis of sperms within the uterus. Now these IUDs are of various types, like it can be hormonal, it can be medicated IUDs, it can be copper releasing IUDs actually. All right, so we can say this is going to match up with C. So IUDs increases phagocytosis of sperms. In uterus. All right. Now the last is lactational amenorrhea. Now lactational amenorrhea is a very natural method of contraception. This comes under natural method of contraception, where it is based on the fact that ovulation and also the menstruation that doesn't take place during the period of intense lactation following parturition. So there is complete absence of menstrual cycle also, and there is complete absence of ovulation also. If a lactating mother is feeding the baby, and it will be following parturition. Following parturition basically means just after the delivery. If the lactating mother is feeding the baby, then this basically chances the reduction of undergoing any menstrual cycle or ovulation. So definitely this is with the D option. All right, so we can say that lactational amenorrhea is a natural method of contraception. All right, so this is a natural method of contraception that has these effects. So if you just match up with the different options that has been given over here, so A is going to match up with fourth, okay, then B is going to match up with first, C is going to match up with second, then D is going to match up with third. That means option B is the correct answer. All right, now let's talk about the next question, which is question number 99. Now the question number 99 says that which of the following is a correct statement. Now this question, if you just read out the statement, the first statement says that autotrophic organisms classified under kingdom Monra, this statement has been actually a little reversed. So kingdom Monra contains cyanobac, or this statement has actually been a little mismatched actually. So basically it wants to indicate, or it wants to suggest that kingdom Monra basically contains one of the category of cyanobacteria, which are a group of autotrophic organisms. All right, so the next is bacteria are exclusively heterotrophic organisms, then slime molds are saprophytic organisms classified under kingdom Monra, and mycoplasmas have DNA, ribosome and cell wall. So this is a question of your biological classification chapter from your 11th standard. All right, now let me tell you that the first statement which says over here, and you have to identify the correct statement, remember. So the first statement which says that Kingdom Monera, see, Kingdom Monera basically contains, let me tell you, that Kingdom Monera contains bacterial species. All bacteria contains bacteria. And in case of bacteria, there are basically three classifications, three important classifications that are there. The first is photosynthetic autotroph. Photosynthetic autotrophic bacteria. Then chemosynthetic autotrophic bacteria. Autotrophs. And the third one is the heterotrophic bacteria. These are the three categories. Now, the cyanobacteria, which here it is talking about, this comes under this category only, photosynthetic autotroph. Photosynthetic autotroph. All right, so this is definitely the correct statement that we have found out. All right, so the first statement has turned out to be the correct statement, but let us take a look into the other statement also. So the next statement says that bacteria are exclusively heterotrophic organisms. So definitely this is not the correct statement. This is completely incorrect because we have seen over here that there are three categories: photosynthetic autotroph, chemosynthetic autotroph, and heterotrophic bacteria. And let me tell you that here I am talking about the eubacterial classification, eubacterial categories, eubacterial categories. Because bacteria also has two categories, eubacteria and archaebacteria. So I am here talking about the eubacterial categories. All right. So this is incorrect because they are not exclusively heterotrophic. This word is wrong. Now, slime molds are saprophytic organisms classified under Kingdom Monera. Not at all. This is also incorrect because slime molds are included in the Kingdom Protista. So we can say that slime molds are saprophytic protist. So since these are saprophytic protist, so they are classified under Kingdom Protista. Classified under Kingdom Protista. All right, so this is also incorrect. And the last says that mycoplasmas have DNA, ribosome, and cell wall. All right, now let me tell you that mycoplasmas are the smallest living cells known. All right, so mycoplasmas, they are the smallest living cells known that can survive without oxygen, but they lack cell wall. They might be having DNA and ribosome, but they lack cell wall. So mycoplasma lacks cell wall. That means this is also incorrect. So our correct statement is option A over here that we have found out. All right, now. So the last question of this test is question number 11. Question number 100. So question number 100 says that given below are the two statements, and statement 1 and statement 2. Now with regard to the light of the above statements, you have to choose the most appropriate answer from the options given below. Now let us take a look that which statement is correct or which statement is false. Now statement 1 says that in a scrubber, the exhaust from the thermal plant is passed through the electric wires to charge the dust particles. And statement 2 says that particulate matter, which is 2.5, which is having a size of 2.5, it cannot be removed by scrubber, but can be removed by an electrostatic precipitator. All right. Now, this question is basically the question from environmental issues chapter of your 12th standard. All right. And this statement, the first statement which says that in a scrubber, the exhaust from the thermal power plant is passed through the electric wires to charge the dust particles. Now, let me tell you, scrubbers are basically used, by those industries that produces sulfur dioxide as a product. All right. And this process of using electrical wires is not in the case of scrubbers, but it is in the case of electrostatic precipitators. These are not in the case of scrubbers. All right. So we can say that statement 1 is considered to be as actually wrong. So statement 1 is incorrect. Statement 1 is considered to be incorrect because scrubbers is used by industries that produces SO2. That means sulfur dioxide as a pollutant. All right. Now, the limestone which is actually present in the scrubber, this is going to remove it from the exhaust particle, from the exhaust of the thermal power plants. All right. So here there is no requirement of any kind of electrical wires or passing out the process. So the limestone found in scrubbers removes SO2 from the exhaust of thermal power plants. So that is the case. Now, what about statement 2 now? Statement 2. So if you just look into the statement 2, so statement 2 says that particulate matter, which is PM 2.5, it cannot be removed by scrubber, but can be removed by an electrostatic precipitator. Now, this is considered to be as a correct statement. This is a true statement because electrostatic precipitator is one of the most effective devices that uses or that can rather remove 99% of the particulate matter that is found in the exhaust, and even if it is of size 2.5. All right, so we can say that this is correct statement as electrostatic precipitator removes 99% of SPM, even it is of particulate matter size of 2.5 present in exhaust. All right. So that's why we can say that option, that means statement 2 is considered to be as the correct statement over here. All right, so let us take a look that what are the options that has been given. So both statement 1 and statement 2 are correct. No. Then both statement 1 and statement 2 are incorrect. No. Both statement 1 is correct, but statement 2 is incorrect. Again, no. Then statement 1 is incorrect, but statement 2 is correct. So this is the correct answer. That means option D is the correct answer over here. All right. So this is all about the questions of your NEET 2022 test series of the biology, and this is how we have completed the entire test through by solving all the questions of biology in details and in depth. All right. So that's all. So, till then, and we will be coming up also with a lot more number of questions and lot more number of papers as well. So till then, keep learning and keep scoring high. And for any kind of doubt related queries regarding this paper or something, you can join our Telegram channel link, which is given in the description section below. Thank you very much.